Good afternoon. Everybody. Am I audible?
Yes, ma'am, you are audible. Yes.
Yeah, so I request all of you to mute yourselves. So this is an honor and pleasure. So on behalf of School of Chemistry, School of Medical Sciences, University of Hyderabad, would like to welcome everyone for this August meeting of celebrating the Indian Women Scientist Association's Golden Jubilee year. And we are honored that we have been given this proud privilege to start the first event of the Golden Jubilee celebrations. And I especially thank our Vice Chancellor for sparing time and supporting this program. This program has multiple speakers, and it's not that I just wanted to invite only women faculty. I know all scientists are there. I'm general neutral, but this is a golden jubilee here, and I have had occasions where I had to hear that in a woman scientist meeting, the team member, the male member present. So I remember that comment. So I made this a special effort to showcase the effort of all our women scientists. But this by no means, it suggests that we only are the scientists. As I said, I declare being gender neutral in terms of scientific research. And I'm a clinician scientist. And in this forum, I just would like to talk about few words that we keep hearing every time about glass ceiling, imposter syndrome, the leaky pipelines, then the ivory towers, and so many things. But despite all that, and the work balance, work-life balance issues, so what? Nothing succeeds like success. And the very fact that these scientists are presenting here, it only goes to show what success can do. So this is only a glimpse or a tip of the iceberg of all the contributions of women scientists, and I definitely we cannot cover everyone. Those who got dropped, it's by no other reason except that these people seem to fit the theme better. And Dr. Lakshmi and Dr. Ratna had asked us to submit our concept notes, and being from and the entire society is interested in this program.
Yes.
So
Audio
therefore,
for a few seconds.
oh, I'm sorry. So
Again.
was the theme of advances in technologies in health sector. And that's the reason some of you were chosen specifically for your great contributions. And with these few words, all the words about leaky pipelines, glass ceilings, ivory towers, all keep them aside. Just enjoy the progress of women scientists in this meeting. A very special thanks to our Vice Chancellor, the IWSA members, the entire team of IWSA from headquarters as well as Hyderabad chapter for giving us this opportunity. And on behalf of Lalita, my co-host from School of Chemistry, we thank all of you for joining us. And I would now request Dr. Ratna to talk about the IWSA, Hyderabad IWSA proceedings, and then followed by Golden Jubilee celebrations by Dr. Lalita, and followed by welcome address by our Vice Chancellor. So once again, welcome to all. Thank you everyone for cooperating in preparing this program. Thank you.
Okay, I'm Ratna here. Good afternoon to all the people present here, all the dignitaries, particularly Professor B.J. Rao garu, Vice Chancellor, University of Hyderabad, the eminent speakers of this day, the team from IWSA headquarters, members of IWSA from our Hyderabad, as well as all the other branches, dear colleagues, other scientists, students, and all those present to the meeting. A warm welcome for this event. I'll talk a bit about IWSA. This IWSA, Indian Women Scientists Association. Now today the celebrations for 50 years have started. This is basically formed for service to science. It is headquartered in Mumbai, and currently it has 11 branches across India with a strength of more than 2,000 members. Its main objective is science to society and encompasses events from school level to research, from common man to experts in various areas of science, and from rural to urban areas. This is a forum which captures a rich resource pool of women scientists dedicated to serving the society through knowledge sharing, mentoring, and motivating girls to take up science as a career. It is on a mission to create a platform to bring all the women in science together and contribute substantially to popularizing science and work towards creating role models in science for the young students to emulate. IWSA also undertakes a lot of community programs like healthcare centers, daycare centers, nursery schools, scholarships for students, working women hostels, etc. In this manner, they have been able to touch lives of numerous people in this journey of 50 years. And also, IWSA collaborates with a number of government as well as non-government agencies to conduct all these events. IWSA membership is open to not only people with scientific background, those in science, but also those who are interested in science. Now coming to the Hyderabad branch, it was formed in 1977, and we are actually proud to be part of this group, which was started by 12 women scientists way back in 73. Right here we have Dr. Mehta Bamji, who was one of the founder members of the IWSA Hyderabad branch and has been an inspiration to many women scientists and is still an active member. And we see today here, and it's always inspiring to be in her presence. IWSA Hyderabad grew slowly over the years. However, this small group of scientists were very dedicated and conducted some very big events, including a recently held national triennial conference, which attracted more than 300 women scientists across India. So at this juncture, the branch is poised to grow into an association of women in science, which could make a huge impact on the society. So we are happy to inform you, as Dr. Geeta has already said, that this is the first event in the series of events to be conducted as part of Golden Jubilee celebrations of IWSA. And we thank the headquarters to have given us this opportunity. Of course, I should mention here, the entire credit for organizing this event goes to Dr. Geeta Vemuganti and Dr. Lalita Guruprasad of University of Hyderabad, who have organized this event in a very short span of time with a galaxy of eminent speakers in this theme that we have chosen, Advances in Technology and Health Sector. It is an apt and relevant theme in the present context. We thank all speakers who have agreed to be part of this event. We are sure the audience will gain immensely from these talks. On behalf of IWSA Hyderabad, I wish the event a grand success. Thank you.
Thank you very much, Ratna. As I said, the pleasure is ours. And the keynote speaker, as I just mentioned, is Dr. Sharma Bhattacharya. He needs a special thanks because she's going to get up at 4:30 a.m. to join us, and she accepted our invitation at a very short notice. And she stretched. In fact, this night for her, she gave a mock drill and confirmed that she's all set, and she's going to get up at 4:30 to join us. So a special thanks to her.
Yeah, please convey my thanks to her. Over to Dr. Lalita Dhareshwarai. Ma'am, you're muted.
Yeah, good afternoon and a warm welcome to all the eminent speakers, our Vice Chancellor of University of Hyderabad, and all the participants for this important webinar on advances in technology in health sector. This webinar is important for two reasons, as already Ratna has mentioned. Firstly, as I glance through the schedule, I'm seeing talks which are very exciting and in the frontier technologies. And second one is that we are launching our Golden Jubilee celebrations programs at the branches with this webinar organized by IWSA Hyderabad branch. As just now you heard about IWSA, what is the speciality of this particular organization? It has 2,300 members from various disciplines of science and technology who can contribute enormously for our main mandate of science education and taking science to masses and make a significant difference to our society. The Indian Women Scientists Association will enter its 50th year on 13th of June 2022, and it was founded in 1973 on that day. We would be ushering the Golden Jubilee year with a grand celebration of Environment Day on the 13th of June 2022. Now, if we look back, 50 years is a very long period through which this organization has been very lovingly and very, very in the hard, by the hard work of several people, it has been sustained. And we members at the headquarters and branches have tried our level best to realize the dreams of the 12 committed founder members. If at all, the activities have grown enormously. They have increased over the last few years. And the most important thing is that IWSA has been resilient through COVID, running the Working Women's Hostel at Vashi, Navi Mumbai throughout this period. There have been more than 100 lectures and webinars online, new programs under Vigyan Prasar, internship programs and programs connected with our unique learning garden. So IWSA has come a long way and made its presence known all over India through its 11 branches. Out of the 2,300 members, I can easily say about 200 members are very actively involved in the various activities of this organization. And I'm sure after this webinar and in this Golden Jubilee year, we will have maybe double or triple of this numbers. So we are carrying out our various activities under our four main mandates, that is taking science to masses and bringing scientific temper in society and promoting the accomplishments of women in science and technology, which is in a way empowerment of women, and to be a representative body of women working in science and technology. These are only the four mandates. There are four more mandates which refer to the various activities that we have taken up. Keeping this in view, we decided to launch the GJC programs from January 2022 itself, because the plans, we have planned for so many programs and 1 year may not be enough. And the first one that we did was on the 2nd of January, that is a logo launching of the Golden Jubilee logo, which is seen on the brochure of the conference. And the tagline is "Look back to move forward and to soar." And this has been designed with a lot of thought behind it. And I would just like to present that logo to you so that you can see it well and understand what is actually the reason and what are the different things stand for there. Are you able to see my screen?
Yes.
Yes. So this is our Golden Jubilee logo, which states very clearly that IWSA is celebrating 50 years of its service to society through science. And the tagline, "Look back to move forward and soar," is a very, very important thing. That is, if we have to move forward, we have to look back work on our experience, look back at our successes and failures, and go ahead how to improvise on these things. So the swirl of colors below shows the winds of change, the yellow and blue swirl of colors. And even in that, you will see it starts with golden yellow going to blue. So as you know, the photon energies for yellow and blue is in the increasing order, which shows that as we move ahead, we are going to move ahead with renewed energy, and we are going to go much ahead than what we are today. And the flying bird represents IWSA, which is using this winds of change. Actually, these waves are indicating the winds of change. And every change, every wind of change, every wave has been used by IWSA to soar ahead, to use it as a springboard. So this is the meaning of the Golden Jubilee, which I thought all of us should know. And using this is a very encouraging and very inspiring logo, at least I feel, and a lot of thought has gone behind it. And in the Golden Jubilee year, the following programs have been planned to take place in the near future. A mural competition has been launched for the students of architecture. And the aim of this was to integrate science and art and architecture and to bring their beauty together. The topics on which the murals had to be designed was the Indian women who broke the glass ceiling, ecological restoration, and women in science. There were 14 submissions from all over Maharashtra, and out of which 8 were chosen for the second round on the 14th of January. The final round of judging is tomorrow, that is 26th of February. We are writing the history of IWSA for the last 25 years also. And apart from this, all our usual lectures in popular science lectures, Science and Our Life series lectures are going on as usual. 25th lecture of Science and Our Life series is scheduled for 9th of April. And we have the woman scientist from NASA, Dr. Hashima Hassan, who would be delivering a talk on unlocking the secrets of the early universe with James Webb Space Telescope. She is the Deputy Program Scientist at the James Webb Space Telescope, Program Scientist of New Star WMK Laboratory, Observatory, Discipline Scientist, Astrophysics Data Archives, Astrophysics Division Lead, and Executive Secretary of NASA Astrophysics Advisory Committee. It's going to be really interesting, exciting when she would be displaying some of the results on the distant galaxies captured by this telescope. So please do join us for this lecture at 6 p.m. on 9th of April. And another important incident is that UNESCO visited our campus on 23rd, that is day before yesterday, to write about our organization and interviewed our founder members, Dr. Sudha Padye, and Chairperson of Board of Trustees, Dr. Sunita Mahajan. Coming to the point as to how IWSA is now involving itself in the important goals set by like-minded institutions. For example, the goal of restoration of ecosystem by UN within the decade on ecosystem restoration aims to prevent, halt, and reverse the degradation of ecosystems on every continent and in every ocean. It can help to end poverty, combat climate change, and prevent a mass extinction. We at IWSA have strived to work in this direction. For example, our mangrove restoration project, in which our Kolhapur branch is also involved, we have started internships for college students to work on such projects. How do we upgrade our knowledge system? By creating a wisdom bridge, which connects one generation to the next. That's what we are trying to do with these young students. We have already completed internships for more than 100 college students, and IWSA has been pursuing work on some of the 17 goals set by UN for transforming our world. For example, community development, skill development, health, etc. Our founder members thought ahead of time, in fact, to upgrade our knowledge systems. And further, we have been trying to integrate arts and science. We are trying to plan and to bring artists and scientists so that one can actually see the beauty which lies behind these two, which are actually, they come out, they emanate from the same source. The Golden Jubilee Celebration program at branch level is being ushered by IWSA Hyderabad branch as we talked just now. And this is the first webinar, and it is covering some of the most advanced and modern concepts such as AI, robotics, space station biology at NASA, from NASA, which are really very, very interesting and exciting, including the topic of the development of vaccine. We would be having equally exciting programs now every month by the different branches of IWSA, not just webinars, but actually reaching out to underprivileged communities through health camps and other means, as is being planned by IWSA Kalpakkam branch. Therefore, I wish all the best to all of you, to all the members of IWSA and today's webinar, the speakers as well as participants, and thank you very much. Thank you.
Thank you very much, Dr. Lalita. It's very inspiring to hear about the history of IWSA. There's no doubt about it. We all should be very proud of it. And with that, I thank you very much. So I would like to invite Honorable Vice Chancellor, Professor B.J. Rao, to give his words and history.
Namaste to all of you. I hope I am audible, visible. Yes, very good. So firstly, this is a very special privileged occasion for me to be part of such a power pack. And being a scientist myself, I really do not feel out of place here at all. So there are no ceilings. We are all in the same boat. This is the spirit that I share. And this is the spirit Professor Geeta Vemuganti also articulated. So I really want to welcome all my visitors, guests from Indian Women Scientists Association. I really want to give my heartfelt gratitudes to the invited speakers of the day, Dr. Sharma Bhattacharya from NASA. I do not know from which part of NASA center she is joining us, so the time frame will be different. Dr. Anuradha ji, Dr. Ruma Sinha ji, and of course, Professor Geeta Vemuganti, Dr. Shilpa Sawant, Dr. Annie Hassan, Professor Geeta Sharma, my esteemed teacher from Osmania University, and Dr. Manjushri Garu, Dr. Sudha Murthy, and Dr. Priya Abraham, Director NIV. So this is a very special occasion. I wish we could invite all of you in person to the University of Hyderabad, but I think this is the next best that we could do now. There will be occasions where we would physically welcome you to the university. So I am actually thrilled to know that this the Indian Women Scientists Association is now 50-year-old. Set up in Mumbai in 1973. And when I stared at the IWSA logo, which I just saw, I found that there is an atomic power right at the core of the logo. So that symbolizes the power with which this organization is powered. I do not know whether it is Mumbai connection, DAE connection, or atomic connection, all put together perhaps, which is very good. The theme of today's discussion is quite profound. That is regenerative. That is actually advances in technology in healthcare. It's a very, very broad topic, and the speakers galaxy that we have here really nicely encompasses various facets of this advances in technology in healthcare. And it would be a real privilege to listen to all of you. Advances in human or medical technology is going through a sea change. As we speak, technology is penetrating clinical care in a very seamless manner. 3D printing and artificial organs, biomaterials, biosimilars, biomimetics, and all sorts of scaffolds, and important problems related to vasculature in the scaffolds, neural innervations in the scaffolds. All these are being tackled so well. And in fact, there are a good number of products available now which are used in a patient-specific manner, which I think is quite an achievement that medical technology has achieved, except that most of these technologies are fairly expensive. So the next challenge is to actually bring this technology down to the prices that most mankind can afford. And I'm sure that will happen in due course. However, the technology growth is rather exciting. So I feel that the IWSA has done extremely well. I was listening to the description that Madam was giving. In some sense, I felt that IWSA has done so well, but it has not reached the nooks and corner of the Indian scientific society very well, I feel. And when I talk to young Indian women researchers, most of them have not heard about IWSA. So it is very important that IWSA should spread out all its tentacles in all its might, so that we can draw a lot more younger researchers and bring in a lot more ideas and put it at a different level. This is my request. And the other request I have is that IWSA portal should change significantly, in my opinion, because there are so many things happening which do not get reflected on the portal, I felt. Because when I heard, the details are a lot more stunning than what the portal is conveying. So these are two suggestions that I want to sort of make it. Hopefully some of these can be taken up. So and another impressive thing I found in IWSA is that it is not only science but society. IWSA is handling both aspects very, very well. Most scientist associations generally concentrate on science. But when I see your activities, it is not only science but society and all sorts of outreach activities related to society, I think, which is very, very impressive and very heartening to see all this. So again, once again, I welcome all of you in this virtual space to the University of Hyderabad. I will sit through some presentations, but I have other commitments later. So at some point, I will quietly sign off, and I'll catch up with all your presentations in a recorded mode later. This is a very, very special privilege for me to be welcoming all of you. Namaste to all of you. Madam, you have to unmute yourself. No, we can't hear you. I can't hear you. There is some audio problem.
Yeah. You're muted, ma'am.
Yeah.
Go ahead.
Good afternoon, one and all. I'm happy to invite all of you to the session 1. I'm honored to invite Dr. Mehta Bamji to chair the session 1. In fact, Dr. Bamji does not require any interaction to the audience here. But just for the sake of completion, I would like to say that she completed her PhD about 60 years ago in biochemistry from Bombay University, and she has specialized in the area of nutritional biochemistry. Dr. Bamji has received several awards. She has published, hundreds of papers. She has also authored many book chapters. But above all, Madam, we are greatly indebted to you because you make your presence felt at all the scientific events, and you're always
audio problem.
And Dr. Bamji to take the floor. Thank you, ma'am.
Thank you very much for the introduction. I am delighted to be here and really very impressed and happy with the topic selected. Congratulate all the organizers, Geeta Vemanguti and all the others. Since the time is very, very tightly scheduled, I would like to invite Dr. Anuradha Shekaran, Director and Chief of the Pathology AIG Hospital. In fact, this is the hospital where I was treated last year. On the application of artificial intelligence in health and disease. This is an area which confuses me, which I'm really looking forward to your talk. So, Anuradha, please.
Good afternoon, everybody. Thank you, ma'am, for the kind introduction. At the outset, I would first like to thank Dr. Gita ma'am, IWSA team, Professor Rao, for inviting me today for this interesting session. And today I would like to talk about the role of artificial intelligence in health and disease. I hope my slides are visible, ma'am.
Yes, yes.
So I would go through the flow of my talk on talking about what is artificial intelligence and machine learning, what is technology innovation in healthcare, role of artificial intelligence in healthcare, a few case studies, particularly in pathology, because I'm a pathologist, and challenges of artificial intelligence, and what is our conclusion for the day. So what is artificial intelligence? Basically, it is also called as machine learning, where the computer systems are taught to perform tasks which normally require human intelligence. So that means the machines are mimicking cognitive functions that humans associate with other human minds, like learning and problem solving. Now, in artificial intelligence, few features which have been performed by artificial intelligence very efficiently and repetitively are speech recognition, decision making, visual perception, and with the advancement of the semiconductor and software technologies, artificial intelligence has been finding many applications in varied fields like self-driving cars, industrial automation, robotics, security, healthcare, and others. So just to know what is machine learning and what is deep learning. Now, machine learning is where the data scientists are going to find out few features which will, for example, if you want to know and identify from the face of the person, the identity of the person, the data scientists are going to teach the machine on certain factors like the roundness of face or the distance between the eyes or the jaw length or the cheekbone structure, etc. And then based on certain algorithms, the classic machine learning is able to come to an answer.
Dr. Bamji, could you please unmute yourself? Thank you.
I am unmute.
That this belongs to a person, Arjun. Now, what is deep learning? Deep learning overcomes these findings of machine learning, and it involves studying of multiple layers of images, multiple neural networks, almost similar to your brain neuron network. And there is a conceptual change from how you engineer the best features to how you guide the model to find the best features. So it trains itself to identify certain features and come to an answer. So the deep learning has seen many breakthroughs in 2010 at Stanford University, where they could come with precision in using this technology in image recognition and speech recognition, which are currently used in your radiology, pathology during the processing, or in the clinical for your case record documents. And also, there has been a lot of breakthrough in the tumor detection, voice assistance, and defect detection. Now, what are the technology innovations in healthcare? In 1950s, there was a lot of innovations in the cardiology, in the bypass, pacemakers, and defibrillators. To 1960s, where we went to the heart valve replacements, intraocular lens, vascular drugs. 1970s saw a sea change of artificial hip and knee replacements, CT, and laser eye surgery. 1980s, we have got the PCR, capsule endoscopy, and laser surgeries and robotic surgeries. And currently, present day, we've moved on to genomic sequencing, the entire whole genome sequencing, the PET scan, which is able to give us an insight into the every nook and corner of the body, the 3D printers, and artificial pancreas. Now, in pathology, what were the breakthroughs? There was not much of breakthrough in pathology except for the modifications in the microscope. But yes, there were three major revolutions. And one was in 1980, where the immunohistochemistry was introduced, which was really a backbone and an important ancillary tool to the pathologist to help to identify whether the patient is dealing with a carcinoma, sarcoma, neuroendocrine tumor, or lymphoma. Because as you know, the pathologist has a very key role to play in giving a diagnosis to the clinician based on which a diagnosis and treatment is made. And immunohistochemistry plays a very, very crucial and critical tool to the pathologist. 2010, the next generation sequencing had become available, which I'll talk in the next slide. And 2018 was when artificial intelligence was started to be used in pathology. So IHC, as we've already said, it is a very important tool which goes to the antigen antibody detection, and the NGS is for detection of the solid tumors, wherein there's a complete gene analysis. But till date, this was done mostly on manual modes, and this was causing a lot of repetitive tasks, which AI has solved many issues in this field. So what is AI in healthcare? If you see that AI has started as early as 1955, and 1960s and 70s saw the use of AI in identifying bacteria and recommending certain specific antibiotics. 1980 and 1990s, there was the generation of this data which was built on the expertise of the physicians. 2010 onwards, there was the role of AI in natural language processing and robot-assisted surgeries, and currently to the personalized healthcare. So the benefits of AI are multifold. So they give access to the real-time data, and this helps in faster and accurate diagnosis. It helps to access expertise from remote areas, saves time and resources, reduces physician stress, reduction in the human error, and it helps to give a more accurate diagnosis. So it gives a reliable monitoring of a patient's progress, and it can be incorporated with your routine reporting, and automatic diagnosis is generated, which incorporates molecular findings, and it helps in research. So as we know that whenever the patient has any symptoms, he goes to the clinician, and based on certain signs, and investigations are ordered, and based on the radiology, sometimes a biopsy is taken, and then based on the pathology diagnosis, the patient is either subjected to no treatment or minimal treatment or aggressive treatment. So in these steps, from the presentation of the patient to the treatment, AI helps in each and every step. And how is it? So this role of AI has seen an explosion in the healthcare, and this is visible in the number of the AI health startups which we are seeing. And AI in medicine market was valued at $719 million, and currently is expected to reach $18,119 million. And also, if you take a look at the number of researchers and the research papers in AI, it has moved from 11,000 to almost 41,000 in the last couple of years. So thanks to the COVID, everything has moved online, and so also is the practice of medicine. And this last 3 years has shown that how telemedicine and telepathology and teleradiology have changed the entire practice of medicine. So in RCPAC, the guidelines have also advocated that they can use these digital pathology forms to diagnose your cases. Now, how is AI transforming healthcare? Basically, AI is a realm of huge data, and it is used in the patient-facing, right from using your chat boxes to wearables and devices to personalized genetics and mental health care, to telehealth from teleconsultations to lifestyle management and disease management. And on the doctor side, in analyzing the medical records, analytics, and medical imaging, and in research for drug discovery, clinical trials, and genetic research. So as we all know, nowadays everybody is having these health trackers and smartphones wherein you're able to monitor your patient's cardiac data, and this is going to help in detection of early cardiac events and even keep an idea on the valvular diseases. AI plays a very important role in ICU patients for identifying the infectious patterns and give a clinical sign to the clinicians even before the patient starts to show any symptoms. So it helps to prioritize tasks that truly require the clinician's attention. In radiology, now radiologists have to deal with multiple and large volume of images, and they're expected to screen the images that at speeds which were previously unheard of. With use of AI in radiology, radiologists are able to view the flat images which are of priority, and they can speed up their reporting, and this improves the patient care. Deep learning networks in radiology helps to isolate particular areas of interest, and in the next generation radiology, it is giving such precise and detailed data that you can completely avoid taking biopsies and thereby minimize the risk to the patient. And also, AI helps to monitor the tumor residual disease, and it helps in comparison of the older images with the current one, and provide a higher level of consistency with unmatched speed. The most important role of AI in in the medical care is in the electronic health records, which we call big data. Now, the data is obtained from your radiology images, pathology images, from your medical records, health records, from your investigations, and from your research. And this is a gold mine of patient data, which is very, very important and crucial to digitization and information. And AI algorithms are based on this gold mine, wherein it is used to evaluate an individual patient's record and come up with a predictive score to assess the risk of a disease based on the previous information and family history. So, to explain the role of AI in pathology, I would like to start with a case study on prostate cancers. Now, whenever a patient has an elevated serum PSA levels or some difficulty in increased frequency of urination and approaches a urologist, who then decides that yes, he is a candidate who requires a biopsy. Now, as we know that when there is a suspicion of cancer in prostate, multiple biopsies are taken from the different lobes of the prostate, and almost these are called sextant biopsies. So the pathologist gets almost 32 slides to evaluate for one particular case. And then the role of the pathologist is to look for certain common patterns, morphological patterns, which are around 5 patterns, and identify the most common pattern, which are called the Gleason's pattern, and give a score of the 2 most common patterns, which is called the Gleason's score, based on which a grade group is given. Now, what is the importance of giving this grade group? This is for the risk stratification, because if a patient falls under the low risk or the intermediate risk, he is offered radiotherapy with androgen deprivation and active surveillance. But if he falls under the high risk group, he is a candidate for radical prostatectomy. And if you have a closer look at the intermediate group, there are again 2 groups here, that is the grade group 2, which is a favorable group, and a grade group 3, which is unfavorable. But if you see the score, it is same, 7. But there's a difference in the morphological pattern, 3 plus 4 versus 4 plus 3. So this is where the pathologist plays a very crucial role in identifying and subclassifying this to determine whether the patient falls under the favorable group or the unfavorable group. Now, just to show for an example, a grade group 3 is where you have discrete glands, and 4 is where you have fused glands. That means there are such subtle features which an expert pathologist has to pick up and identify whether the patient has predominance of 3 or a predominance of 4, wherein we are going to change the Gleason's grade group. And also, the pathologist has to screen the entire length of those 16 course of biopsies which I have shown, and quantify the tumor and give a percentage of the tumor, so that we can come to an accurate diagnosis. And also, the pathologist has to give a complete detailed report whether there was any extension beyond the prostate into the fat or into the seminal vesicle, or whether there was vascular perineural invasion, which are key roles to determine whether the patient has to undergo further surgery. So, many studies have shown that there's a lot of inter and intra-observer variation in identifying this Gleason's score 6 and Gleason's score 7, and this might lead to suboptimal patient management. And there was a study wherein even ancillary techniques like IHC was used, and a study was done on 9 different pathologists in a single institute, and they found that there was a lot of discordance among hospitals and among varied hospitals. And the reason was, it could probably because of lack of expertise on part of the pathologist. So probably an expert review will help to overcome this discordance. And then once the patient is given a diagnosis of cancer, then he is subjected to surgery. And look at the volume of slides which are generated, because we have to sample the prostate in its entirety. And this will lead to an explosion of slides and blocks. And if you take as a single institute in our hospital, you can see that the number of slides were 2,700 to as much as 26,000. So just imagine the amount of physical storage of the slides and blocks which require in giving a precise diagnosis to a patient. So how do we go about? And also the challenges are the number of expert pathologists available are in shortage all over the world with the ever increasing workload. And if you see in India, there is a severe lack of experienced expert pathologists who are less than 10 per million. So this is where digitization with computational pathology plays a very important role, wherein a high throughput scanning devices are used to capture the entire whole slide image, and this will help in quantitative assessment, minimize the physical storage of slides, and it increases the focus on precision medicine as it transforms the diagnostic surgical pathology by integrating the image analysis and machine learning. So AI in pathology helps in diagnosis, prognosis, theranostics, and teaching. And so this is the same example of the prostate cancer, wherein if you use artificial intelligence, it is able to identify this red area is the Gleason's pattern 4 and this yellow area Gleason's pattern 3. So it is able to come up with the percentage of the tumor and accurately analyze. So it is reproducible, it is accurate, and it helps in reducing the workload and thereby reduces the stress on the pathologist and gives them time to focus on areas which require more cognitive work. And another area is where there's discordance between the gastroenterologist on the endoscopic appearance and the pathologist on microscopic appearance, wherein a gastroenterologist, after using the artificial intelligence, is able to say with confidence that yes, this is a small 3 millimeter lesion which is an adenoma. But the pathologist might only identify it as a normal mucosa. This could be either because the gastroenterologist has missed this lesion because it's very small, or
you have 2 minutes. You have 2 minutes.
The pathologist has missed the actual area. And the role of the pathologist in the neuroendocrine neoplasms, where obviously Jobs cancer, wherein we have to grade the tumor based on Ki67. Normally we do it on manual mode, wherein we have to count around 2,000 cells, and this is not feasibly practical. So this is where AI plays a very important role, because based on the AI, the Ki67 grade 1 is kept on observation and grade 2 and 3 go for further treatment. AI has helped a lot in the immuno-oncology and the treatment in precision medicine, wherein it is able to identify certain specific biomarkers, and the goal is to use these genetic and molecular data to build up biological profiles. One such area is the using of PD-L1 for identifying that subset of patients who will respond to a drug called pembrolizumab. And here again, if you see, a pathologist has to count manually the number of viable cells and also count number of lymphocytes, macrophages, etc., and then give a tumor proportion and a combined proportion score. So AI plays a very great role in definitely scoring these, and this is widely seen in the number of the medical drug companies which are producing these drugs. So AI also helps in research, wherein it helps in recruiting and speeding up the processing and cleaning of the data, and to identify the biological targets. The challenges are the development costs, reluctance among the medical practitioners to adopt introducing the AI for fear that it might replace humans. There are no standardized workflows available, as particularly in pathology when compared to radiology. We don't have internet facility at all conditions, and there is this financial obstacles. And the questions still to be resolved are, who is legally responsible for using this AI? Who is accountable for actions resulting from the AI and for the error made through use of AI? But what we need to learn is, AI will not replace doctors, but it will assist doctors, and human element will still remain an important factor. To conclude, AI is part of future in pathology and clinical practice. It's still on the edge of its potential, and healthcare is hope for AI because of big data. AI increases the diagnostic accuracy, improves workflow, decreases cost of human resources, and improves therapeutics by performing repetitive tasks very quickly. So it gives a very precise and predictive clinical decision making, helps in synergistic collaboration between the medical, oncology, radiology, and pathology so as to integrate morphology with genomics, proteomics, bioinformatics, and computer algorithms. Human oversight will still, however, be needed to verify the accuracy of AI outputs and ensure optimal treatment. Thank you.
Very much for this very interesting presentation. Is the moderator Shailaja around? Is she there?
Ma'am, could you please do the moderation?
Let's now invite Ruma Sinha. Can you hear me? Dr. Ruma Sinha, who is the consultant gynecologist at Apollo Hospital, she will speak on advances in robotic surgery. Ruma.
Big namaste to all of you. Good afternoon. It is indeed a pleasure and a privilege to be on part of this August forum of IWSA, which represents Indian scientists, and glad to be part of a scientific community today. Just being a clinician, we always think that we are clinician and working doctors. When Geeta called me to make this presentation, it was an opportunity for me to discuss the advances in the future of surgery that we have actually been witness to and probably be part of it. So I'll share my screen. Let me just start that.
So while Ruma gets prepared for her talk, just wanted to add few lines. You can hear me?
It says Google, I can't share my screen. Anybody can help me with that?
You have to get a co-host.
I probably need to be a co-host or something.
Yes.
Is there somebody from the technical team who can help me?
We're just doing this.
Okay.
Below there will be squares where there is this, in a small rectangle there is an arrow. You
I know.
press on
When I
that.
put that, we have three options: your entire screen, a window, and a tab. Which one do I
You
select?
say the part of it.
Come again?
Part, not the entire screen.
A window?
A window.
Okay. Okay.
Then you
So
say
I need
share.
to, yeah, if I share a window, it says I can share my PowerPoint.
Yes.
But then when I share my PowerPoint, it says Google Chrome might not have screen recording permission on your computer.
I don't know.
All right.
Arun, show the entire screen. Keep your presentation ready.
Yeah. I got it. I think I had to give some permission here. That's good. All right. I think I'm going to do it now. Is my screen visible now?
No, ma'am.
Still no? Sorry about this. I've already given a permission to come back here.
I think it's better if people send their slides earlier and let the moderator show the slides.
Yeah, I'm sorry about this. I am not used to this platform too much, although we have been making many presentations on Zoom. This is a new platform to me, so I'm sorry about this.
It may take a few seconds.
Can somebody guide me through what is my issue? I am not able to.
Ruma, after you share the screen,
Yeah.
you need to click on the presentation button.
Can we go to the next presentation and she can come back?
Okay, you can go ahead with Geeta's presentation and give me a minute in the side. Somebody can help me
And
how I can.
in the meantime, email your presentation to Geeta.
That
And
might
she
be
can-
slightly difficult, ma'am, because there are
Did you
Geeta, what shall we do? Shall we go to the next presenter or what would you like us to do?
Can you hear us? Ma'am, did you open your PowerPoint presentation?
Yes, it's open on my desktop.
Because time is running out.
I understand.
Already 10 minutes have almost passed.
It opens, but the minute I say it is, I want to present, it says that I don't have permission.
Who is moderating? Is Geeta Vemanguti moderating? Well, who is the technical person behind this?
VOH is moderating and we have given Ruma ma'am permission to share.
Oh. But in that case, shall we go to the next presenter?
Please,
Yes.
please go ahead with Geeta's presentation while I'll figure this out.
Geeta, are you ready with your presentation?
I just called Geeta. Yes, Geeta says that she will start her presentation.
I'll be starting now.
Ruma, can you send us your slides?
You send your PowerPoint to them by email.
Yes.
So that they can do the presentation for you. I always prefer to do that. Does she have your, yes, you have their email ID, no? You send your presentation. And then, Geeta, maybe you can start, please.
Yes, I'll be starting in a minute. Can you see my screen now? Yeah. Good afternoon, everyone. It's a pleasure to be the host as well as a speaker. And Dr. Bamji, thank you very much for chairing the session. And I would like to talk about an area that is of interest to me, and that is stem cell biology and tissue engineering. I'm going to talk about the general introduction because of the mixed audience. I'm going to introduce the term of stem cell, the potential of stem cell therapy, tissue engineering, 3D printing, induced pluripotent cells, and then personally my experience in ophthalmology. So there are a few facts of life, and we need to accept that we all start our life from one cell. And the entire growth or the developmental biology tells us what happens to each cell. And if we follow the developmental biology, we would be able to understand stem cell biology and tissue engineering better. So from one cell, the entire fetus is formed, it grows, then we go through the process of the growth, aging, regeneration, and death. And with emerging, despite the emerging technologies, many of the disease conditions still cannot be cured. And with increasing burden of aging, we are going to have more problems of disability and degenerative changes. So how well prepared are we to address this? So I go back to the basic understanding of the hierarchy in the organization of an organism. The cells are the fundamental units. The group of cells makes a tissue, group of tissue make an organ, and group of organs make a system. So from the cell to the tissue to an organ, there is an organization of multiple cells. So if we need to go to replace, if we go into regenerative medicine, we need to do exactly what the nature does of building up the tissues and or an organ. So for that, we need multiple types of tissue. And I usually compare it with building of a building or any structure. You need cells, that is the bricks. You need cement, that is the extracellular matrix or the scaffold on which we grow the cells. And then we have a plan of what to make. The Tom Cruise movie shows is nothing but the face mask of a 3D printing of a face. One more fundamental principle that we generally forget is all the types of the cells in the body, right from head to foot, can be divided into 3 types of cells: the labile cells, stable cells, and permanent cells. The labile cells are the ones which keep dividing throughout our body. Like that means the skin keeps dividing, the bone marrow keeps dividing and produces the RBCs, WBCs, and platelets. The stable cells are the one that means they regenerate very well, and they come to our rescue whenever there is excessive damage, say like liver, bone, and other mesenchymal tissues. The permanent tissues, we believe, they are post-mitotic. And stable cells. This distributes the cells which can regenerate to some extent, which cannot regenerate. So we are worried about the cells which cannot regenerate for themselves or with limited scope. So based on that and based on the developmental biology, the stem cells are divided. They are into totipotent stem cells, pluripotent stem cells, multipotent. So you have embryonic stem cells with different pluripotency. Then you have adult stem cells, the fetal stem cells, cord blood stem cells, and adult blood stem cells. So when I'm saying stem cells, what exactly defines a stem cell? It is an undifferentiated cell which is capable of self-renewal and differentiation. And the adult stem cells are located in all this tissue that I just mentioned to you. That means they are in the labile cells, labile tissues, and the stable tissues. They are in the skin, in the digestive system, in the epithelium of all the cells, and so on. So they constitute about 0.05 to 5% of all cells. From the cell cycle point of view, these stem cells have an asymmetric division. These cells have an asymmetric division in the sense that normally the cells divide in this manner. On the right side is an asymmetric division where only one stem cell is pushed into a differentiation pathway, whereas the other goes into another fate, that is a self-renewal. This is asymmetrical division, is a unique property of stem cells. And there is asymmetric DNA degradation, segregation, low mitotic activity, error-free proliferation, And another thing is it is an underbunker army, what I call it. It's in a quiescent microenvironment or a special niche which protects these stem cells. So these stem cells are governed both by intrinsic factors as well as extrinsic factors. So this is the summary of the adult stem cells, which I will be mentioning in a few ways. They are in the hair, intestine, dentistry, and all. So I'll be just sharing with you one by one. And while we go from cells to tissue engineering, which is now almost catching up with the progress of stem cell, what is the difference or the induced pluripotent stem cells? Any cell in combination with a scaffold or a material, which could be either a biomaterial, it could be 3D printed or a decellularized tissue, and then you add some molecules to it or the growth factors to it. When all these three are combined, then we call it a tissue engineering. And we have a pattern or we have a formula on which we need to devise this. So what do we require? We require materials which are biocompatible, which can accommodate these cells, which can take the shape of the target tissue that you desire, and the required biomolecules based on the tissue, and so on. So the fundamental tissue engineering principles remain the same, whether you make a heart, whether you make a ear pinna or so. So the induced pluripotent cell, unlike the adult or the fetal or the embryonic stem cell, and as we all know, this was the scientist got the Nobel Prize, Yamanaka and Gordon. So OCT4, SOX2, c-MYC, and KLF4. If these 4 genes are added to the cell, then this adult cell goes into a pluripotent cell. And from here, it can make all the other cells like an embryonic stem cell. Now, can they be differentiated? Is there a proof? Yes, there is a proof. This is with the permission of my colleague, my friend, Dr. Masayo Takahashi from Japan, right?
Why are you getting suddenly muted? In between we are not able to hear.
Hear me? Yeah. So I was coming to the different types of adult stem cells. There is the progress in cardiology, dentistry, and in different fields. So here is something that is very exciting in the field of vascular diseases. The scaffolds can be tubular. We can line them by endothelial cells because for any vessels we need endothelial cells, and they could be derived from either another endothelial cell that is umbilical or by bone marrow cells. And the cardiac valves are being made by the process of tissue engineering. The vascular regeneration is being tried by various methods, and endothelial cells are derived from different. So this, even in there is a lot of efforts to make vessels and cardiac valves, which could in future probably replace the artificial valves. These artificial valves have a lot of blackouts. So therefore, biocompatible, we can prevent clotting is something that we should look forward. What is going on in pancreatic cells? They have stem cells, they have been derived from ES cells, from bone marrow, from pancreas itself, and now the pancreatic is present. The pancreas, the pancreatic cells are being removed, and then finished cells are being reinfused to the patient, especially in cases of chronic pancreatitis. The skin is one of the closest to the sheet of epithelium using different forms of scaffold and transplant it back to the patient. There are now companies which do that. So, but I must emphasize every now and then that all the principles of approved therapy, prohibited therapy, and restricted are given in ICMR guidelines for clinical application. Initially, I said the brain stem cells are, there are no stem cells in the brain. But over the years, especially the last decade, first in the mouse model and subsequently in the human, we now know that the subventricular zone or the anterior lateral ventricles is a site where you could have the neural stem cells. And this neural stem cells, they may not come to our rescue in our physiological state, but it's still in the research phase. The closer to a potential clinical application is that you could make a neural tube or promote the growth of axons into a tube. So these are the efforts that are being made for neural regeneration by the creation of a neural tube. And the neural cells have been attempted for transdifferentiation from ES embryonic stem cells, induced pluripotent stem cells, as well as bone marrow cells. The tissue engineering or the stem cell biology in nephrology is that it's again one of the very complicated cells, tissue, and organ. It's a very complex of a secretory system and of a ductal system. So here the entire kidney they're trying from embryonic stem cells and iPS, but individually the tubular cells have been regenerated from using bone marrow cells, or a biocompatible dialysis, as we know, is one of the, it acts like a kidney, but a biocompatible kidney is being attempted to form. Can we make a bladder? The bladder regeneration again is in the research phase where they are attempting using a scaffold to fill in the cells to make a bladder. Orthopedics, this is another low-hanging fruit. The attempts, there is a company which was made many years ago, which can offer to take a biopsy of the cartilage, expand it into sheet of cartilage cells, and inject it back into the patient to regenerate the cartilage. These cartilage cells can be autologous source or from bone marrow, or using different scaffolds, they can be made into a 3D. They are bone bands and so on. So there is a lot of progress in the form of bone and cartilage degeneration. Liver stem cell biology is again exciting, but with the success of split transplantation and 3D, there is less effort on putting the fetal cells, which had been attempted earlier, of using fetal stem cells to regenerate the liver instead of patients. 3D tissue engineering is one thing which is progressing in the liver, where there is arrangement made of the hepatocytes, of the bile ducts and endothelial cells to make a 3D organ of the liver. The milk tooth, you can be saved by the kid to make the fairy can bless them with multiple gifts. So now this fairy has become a company where they come and collect the deciduous tooth from the fallen tooth, and you can generate stem cells from the deciduous tooth. So there are multiple types of cells from the dental area. So you can generate an epithelium, you can generate a mesenchymal stem cells, and in fact, one of my postdoc is working on the mesenchymal stem cells from the dental source. And P3 imaging can also be attempted. So is it always cells? Do we always need cells, or can we use the secretome of the cells? Yes, there is a lot of progress in the secretome of various kinds of cells. This is an example of mesenchymal stem cells, which can release small molecules, the nucleic acids, the peptides, proteins, and most importantly, extracellular vesicles, which are the carbon of communication between them. This is one of the landmark papers where a cadaveric tissue or the decellularized tissue was used to re-inject the cells and used to substitute tracheal tissue engineering. So hence, since then, the decellularized tissue has been given importance. Endometrium is another source of stem cells for endometrial regeneration. Muscle stem cells, the satellite cells are there, but do not have, there is still progress in happening in this area. Tissue engineering, these are some of the examples of tissue engineering. I now come to the part of our contribution, the first of its kind, where we wanted to treat the ocular surface diseases. There are various kinds of blinding diseases, the ones which in the red, that is the limbal cornea, dry eye, and our efforts for retinal degeneration is what I'm going to talk to you about. We started the same way. How do we start? What are the questions we asked ourselves? Which cells are we going to use? What the white part, the conjunctiva, and the junction is the limbus. So in cases where this ocular surface is damaged, as you see here, because of chemical burns, what the surgeon can do is they can take the limbal tissue from the good eye and transplant it to the diseased eye. But then it could pose a threat to the donor side. So the Italian group and Raysai from US, which was published in Lancet and NEJM, we were not the first groups to start this. They had attempted to take a limbal biopsy from the good eye, made it into a sheet of epithelium, and transplanted to the diseased eye. And that's exactly what I did when we were at LVPI Institute. We took the limbal tissue. I used human amniotic membrane as a substrate. We stopped using the feeder cells. And then, instead of the feeder cells or the culture insert, we tuck the amniotic membrane on a glass like you would tuck a baby at bedtime. And millimeters biopsy from the good eye, transported it. On the amniotic membrane, we made explant cultures and grown it into sheet of epithelium, which showed stratified epithelium. After full characterization of the cultured cells, these were the surgical debridement was done of the diseased eye, and this sheet of epithelium was transplanted, which restored the ocular surface. If it was only epithelial damage, the vision was restored. If it is a deeper stromal damage, the patient required another surgery called corneal transplantation. One good thing about this was we had access to the tissues which underwent transplantation. So therefore, we could provide the proof of the survival of the transplanted cells by histology as well as by clinical proof as seen in this case. So we have published series of papers wherein the 10-year survival has been published. So if the limbus is damaged, cornea is damaged, you do one. What if both are damaged? So this is another novel contribution of ours, where we attempted to make two types of sheets of epithelium on one single membrane using a ring barrier. I used a ring barrier because the limbus is circular. So if you keep a ring barrier with one type of cells in the center and one type of cells outside, you can make a composite picture, a composite sheet of epithelium. Gita,
you have now 2 minutes, please.
Months, the success was less. But if they survived, we could recommend the success of 20% of the graft survival for more than 10 years after transplantation. So clinical proof of survival of the transplanted cells, histological proof, and genotypic proof. All these submitted proof to show that the transplanted cells. What we reported the presence of mesenchymal stem cells that I talked to you about earlier. So every knee has and found in the limbal stroma. But in addition to the limbal epithelial stem cells, the limbal stroma has mesenchymal stem cells. Is it of interest? I think it's more than that, because these stromal cells are now being used to treat the corneal scar. So limbal epithelial cells have been used, limbal stromal cells have been used, Untitled cells have been used. What next? It's like the cartoon of the Reader's Digest, where one pattern of direct transplantation led to cultured cell therapy, which we called it as CLET. If you combine the explant culture, not in the lab, but directly on the patient, that's called CLET. So now many groups are attempting to do direct cultivation on the patient rather than in the lab. And this is successful, where the second method fails. So, what I mentioned here about stem cells as a niche, is niche important? Very much. The attempts of the Japanese group to substitute limbal with the oral, they reported a huge success. When we tried that, we found only 30% success, which was not acceptable to us. And we believe that the oral mucosa does not work on the epithelium of the cornea because the stroma or niche factors which are required are absent in oral mucosa. The diseases, can we do that? We attempted it.
Condition now it's ended over.
So the neural stem cells of retina we were not able to do because they did not show the proof of principle in animal experiments. Last part of that work is the lacrimal. This was a proposal from the Agency for International Atomic Energy, which asked us to think of damage for all surface diseases. Show that we can make clear substances. So the summary of it is the basic research. Some of them go to preclinical phase, some of them go to clinical phase, like we showed in limbal, in oral, and rest are in the pipeline. So I would like to summarize that knowledge of techniques, disease process, the biomaterials, biology, all the principles and the success of regenerative medicine. We need to be in sync with regulations which approve these clinical applications of different kinds of cells. In ophthalmology, and this is in the pipeline for as one of the therapies or research. With that, I would like to thank my students, my colleagues, and everyone else. And thanks to Inka for this.
Thank you very much, Geet, for a very interesting and sharing your practical experience. Now, can we go back to Ruma? Is Ruma ready with her PowerPoint? Unmute yourself, Ruma. Please unmute yourself. Can't hear you. Has she sent her PowerPoint to the organizers to show the? Unmute yourself. If she has been muted, please unmute her.
She's already the co-host, madam. She can present.
But she has difficulty. Why doesn't the organizer show her slides?
Can you send the presentation to us? That's okay.
I told her to send it by email.
Yeah.
Okay, otherwise let's go on to the next speaker and come back to her later. Can we go to the next speaker?
Yeah, she's presenting, madam.
She's got it?
Yes, Ruma's presentation. Ruma, open your presentation. Will we see your emails?
Yeah, my presentation is open on the.
Close this. Close your email box.
Yes, guys.
Is it okay, Ruma? We go to the next presentation. Are you ready?
Go ahead, Geeta. I'm figuring it out. Actually, unfortunately, the presentation is so large. It's about 6 GB with videos, so I can't send it on the mail. If somebody can help me, how I can
Oh,
transport it in some other way, I will share it. If your tech
you,
team can give me another link to share this presentation, I'll do it. But it's not
no,
going on the mail.
there is another method of sending such big presentations, no?
Yes, there is, ma'am, but I just don't know which one your tech team will receive. So if they can send me a mail with a link, I will send it immediately.
So let's go on to the next speaker and then come back to you again, Uruma. Shilpa Samant, scientist, Bhabha Atomic Research Center, to speak on biosensors for healthcare application. Shilpa. Is Shilpa there?
Yeah, I'm there, madam. Can you hear me?
Restart your presentation.
Yes. Good afternoon. Am I audible?
Yeah, you're audible.
Okay. Let's start the presentation.
It's 3:28 now.
Yeah. The slides are visible now?
Yes, right. If you were to, please make it full screen. Go ahead, go ahead.
Yeah.
Please go ahead, Shilpa.
Yeah, just a minute, huh, madam? Just a minute. I think the slides are seen.
What's the problem? Mr. Pallavi is presenting. What's the present? The delay due to? Please start.
You are muted, ma'am.
We cannot hear you. Please unmute yourself.
Dr. Shilpa, we can see, we can.
Yes, madam.
We lost your slides.
Now I think all this is fine. Right? Yeah. First of all, I would like to thank Professor Geeta and Shamla Bharatwaj madam and also the organizers for the invitation for me to be a part of this event.
Unmute yourself. We can't hear you. Something wrong with your audio.
Rest all can mute, please.
slides are the slides visible?
Yes.
Yes. Okay.
Yes.
Okay. Okay. Yeah. Yeah. Thank you. Thank you. Okay. Yeah.
Go ahead now.
Yes. So today I'll share some of our work on biosensors for healthcare application. So I've divided this talk into 5 parts. I begin with an introduction to biosensors, followed by the various parts of the biosensors. That's the biorecognition unit, the transducer, and also I'll present a few examples of the different type of biosensors, like the colorimetric biosensor, SPR based biosensor, and electrochemical biosensor. And towards the end, I'll describe about our work on device development. Actually, we have developed a device for detection of cancer in biopsy samples. So I'll just share some of the details there. Now, see this recent pandemic, it has shown us that there's a need to have a resilient healthcare system. And we have seen how a simple device like an oximeter can help to monitor the progression of COVID. So that way, sensors are very much important in healthcare. In fact, the first talk, Dr. Anuradha also mentioned about all these AI methods where all these sensors can be integrated to have the data together and all, and that can be analyzed. So, but if you look around what the sensors around us, what we see is mostly the wearable sensors, like the activity monitors or the smartphones or the Fitbits. These are actually activity monitors, like they tell us about the number of steps we take or the heartbeats, heart rate and all. So these are like personal metrics which basically talk about the fitness. But many times what we need to know about our health is in more detail, where some details are required at a molecular level. And that is where biosensors play an important role. So biosensors are required for personal monitoring, like at home. Like the glucometer is one of the most popular example of a biosensor. Biosensors are also required as point of care device at the bedside, and also in the case of pathology labs where a large number of samples are handled simultaneously. So in the recent time, one of the most sought after sensor or biosensor is the COVID antigen test. So this is based on lateral flow immunoassay, where antibodies are used, and it is also possible to visibly see this color marks here. But if it is integrated with a reader, then this whole device becomes a biosensor along with the transducer basically. So biosensor is actually a full-fledged device which can independently monitor biochemical signals basically. So it is a combination of analytical biochemistry and microelectronics. It's a synergic combination of these two. It's a standalone device which can do proper analysis and also give the data in the form of a current which can be displayed basically. So this biosensor, it consists of a bioreceptor. Now this bioreceptor could be anything like an enzyme or DNA, RNA or tissue or cells. So this bioreceptor, it interacts with the analyte to generate a biochemical signal. So this biochemical signal is picked up by the transducer. This transducer can be of different type. It could be an electrochemical transducer, optical transducer, a thermal or a piezoelectric transducer. So this transducer, it converts the biochemical signal into an electrical signal which is displayed. Now biosensor find application in various areas like in clinical diagnostic, in food analysis, pharmaceutical, environmental monitoring and all. So today I'll be presenting some of our work on biosensors for healthcare, where we are working on colorimetric sensor for detection of metabolites, bacteria, glutathione and all, and SPR based sensors for detection of cancer biomarkers, electrochemical sensors for detection of metabolites, cancer biomarkers, bacteria, and cancer cells. Now, if we see this, as I mentioned, the biorecognition unit. Now, this unit is considered to be the heart of the biosensor because, and depending on the type of the moieties which are used, the biosensors are divided as enzymatic biosensors, antibody-based biosensors, nucleic acid-based biosensors, and whole cell-based biosensors. Now, the interaction between the analyte and these moieties is highly specific in many cases because it follows a lock and key type of mechanism which gives selectivity to the sensor. Now, this is a typical example of an enzymatic biosensor. So what is done is, say this is a glucose sensor. So to develop a glucose sensor, the corresponding enzyme, that is glucose oxidase, is taken, and then it is immobilized over a electrode using appropriate sensor material, and then this electrode is exposed to glucose. Glucose is converted by glucose oxidase into gluconic acid and H2O2. This H2O2 can be reduced at the electrode to give an electrical signal. Similarly, for detection of other analytes like cholesterol oxidase enzyme can be used. For detection of urease enzyme can be used. So this is one of the examples where we have developed enzymatic sensor for detection of uric acid in blood serum samples. Here, a composite of polyaniline and Prussian blue was used as the sensor material because it acts as an electrocatalyst for reduction of H2O2. So the electrode was first modified with this polymer composite by electrochemical method, and then the enzyme is immobilized using glutaraldehyd coupling. This is the response of the sensor to uric acid, and it shows a linear range in the 10 to 160 micromolar range and shows a good selectivity over common interfering agents present in the serum. Now, instead of using pure enzymes, it is also possible to use whole cells, the enzymes present in whole cells. Or alternatively, one can genetically modify cells to overexpress the required proteins so as to give specificity to the sensor. Like this is an example where we have used E. coli cells which were genetically modified to have a gene called LIN-A2 gene. Now this gene, this can specifically degrade lindane. Now lindane is a insecticide and it is known to be a carcinogen. So it's banned in several countries, but it is highly persistent and it remains in the water, groundwater. So its detection is important. So this gene is able to degrade this into smaller components. Now, what we did is we immobilized these whole cells in the polymer matrix, and this is the response of the sensor at different loadings of the whole cell. So this sensor was highly specific and it did not respond to other chloro compounds. It did not even respond to other chloro insecticides, but it showed a very good response to all the isomers of lindane. Now, this example is slightly off from a healthcare risk, but I just wanted to show it because this is how one can use whole cells and genetically modified cells to design novel biosensors. Now, coming to the second part of the biosensor, the other unit is the transducer unit. Now, as I mentioned, the transducer can be of different type. We are working on mostly optical biosensors, colorimetric and SPR based, and also electrochemical biosensors. I'll just present one example of each one of these just to give a flavor of the topic. So here we have developed colorimetric sensor based on polyacetylene LB films for detection of glucose. Now this polydiacetylene, it has alternate single and double bonds. So this is a rigid polymer backbone and it has a conjugation. And these polymers are known to show mechanochromism. That is, when pressure is applied, there is a disruption in the conjugation which leads to color change. So what we did is we prepared a 3-layer LB film, Langmuir-Blodgett film, which are monomolecular layer films. So we prepared LB films of this and covalently immobilized glucose oxidase. When the substrate binds to glucose oxidase, what happens is it stretches the bonds in the polymer, which disrupts the conjugation, leading to a color change. Now, this color change can be easily seen from the UV visible spectra, and it can be quantified in terms of the colorimetric response. So this is a colorimetric response for the sensor and shows a good response in the physiological glucose concentration range. And the sensor was found to be specific. It responded to glucose but did not respond to the other common sugars. Now this same polydestin system we used it to study another interesting protein substrate combination, and that is enolase protein and its interaction with 2-phosphoglyceric acid. So when the LB film is immobilized with this enolase and it is exposed to the solution of this ligand PGA, there is no color change observed. But when magnesium ions are added to the PGA solution, immediately there's a change in the color. So this is because magnesium ions are essential for the activity of this enolase enzyme. So this way we could trace the enzyme substrate interaction using this type of films. Coming to the second type of transducer, that is where we can use the SPR, that is the surface plasmon resonance principle, to develop biosensor. Now this SPR, in this sensor, what we have is we have sensor chips which are coated with very thin layer of gold so that it can sustain the SPR wave. And on this, we bind antibody using various chemistries to covalently bind these antibodies. So when the antigen binds to these antibodies, there's a shift in the SPR signal, and this shift is proportional to the amount of binding. So this is how it can be used to study various biomolecular interactions like antigen-antibody interaction or protein substrate, DNA-protein interaction in biosensor research. So what we have done is we have tried to develop a SPR-based sensor for detection of cancer biomarker alpha-fetoprotein. So this is a liver cancer biomarker. So we immobilized it on the sensor chip using 3 different chemistries and then tried to study its interaction using SPR. So these are the typical sensograms which are obtained, and this is the response to different concentration of AFE. So this sensor could be used to determine AFE in serum samples. The third type of transducers, that is the electrochemical transducers. These are very popular because they have a lot of advantages. It is because it is possible to make portable device, because these can be miniaturized very easily to have a lab-on-chip type of sensor. They are simple to operate, and also one can analyze multiple analytes using these type of sensors. For developing electrochemical sensors, a variety of transducer materials can be used, like carbon-based materials, inorganic, organic nanoparticles, insulating polymers, biopolymers, conducting polymers. And among these, biopolymers and conducting polymers have been one of our favorite materials. It's because the transducer material needs to be biocompatible. It needs to allow diffusion of the analyte to the bioreceptor, and it should have a good signal amplification and transduction. So what we did is we prepared nanoparticles of this polymer, polyaniline, which is a conducting polymer, and used it to immobilize glucose oxidase enzyme. Now, this nanoparticle-based sensor showed 3 times higher response as compared to the normal polyaniline-based sensor. And it was also used to determine glucose in serum samples. In another example, we prepared mesostructured polyaniline using combination of surfactants, like different surfactant combinations were used, and a set of polymer was synthesized. So here, for this mesostructured polyaniline, we observed almost 8 times higher sensitivity as compared to the normal polyaniline. So here, by controlling the morphology, it is possible to improve the sensitivity of the biosensor. Now, after having discussed a few examples of metabolite-based sensors, I move on to biosensors for detection of cancer, basically cancer biomarker detection. So this is an example of an electrochemical biosensor for detection of alpha fetoprotein, which is, as I mentioned, is a liver cancer biomarker. So what was done is screen printed electrodes were used, and on those silver nanoparticle was deposited by electrochemical method. Then the antibody for AFE was immobilized, and its interaction with the antigen was studied by differential pulse photometry by using redox mediator. So this is a response of the sensor to different concentration of AFE. In another strategy, we modified the electrode with a composite of polymer, nanoparticles, and BSA to entrap the AFE antibody, and this electrode was then exposed to the antigen. And after this antigen exposure, it was treated with this type of detection probe to have a sandwich type detection, where this probe is basically metal nanoparticles on which HRP conjugated polyclonal antibody was immobilized. So in this case, when this electrode is treated with H2O2, H2O2 gets reduced to give a signal, and this is catalyzed by the HRP enzyme. So in this case, having such kind of detection probe helps to improve the sensitivity 3 to 4 times as compared to having a normal detection probe. And this sensor was used for estimation of AFE in blood serum samples also. Now, having worked on several biosensor systems, we selected a few systems which have a potential for real-life application and went ahead to develop device for these. Now, one of the devices is electrochemical sensor for detection of cancer cells. Now, as Dr. Anuradha mentioned in her talk, there is a scarcity of number of trained pathologists to analyze histopathology samples, and also it is desirable to have a simple technique to screen the samples initially, for the initial screening of samples which can be used by technicians. So from that point of view, we have developed a sensor based exploiting the difference in the glycolysis rate of the normal cells and the cancer cells. This is because cancer cells are known to have abnormal metabolism, and they take excessive amount of glucose and have a high rate of glycolysis. So we validated the sensor using cell lines with mice tumor tissues and human tissues. I'll just share some of the results. Like these are the results for the mice tumor xenografts, MCF7 xenografts. So this is the response of the sensor for tumor tissue, and this is the response for the normal tissue. Now there's an increase in the current in the case of tumor tissue because of the high rate of glycolysis. Now to confirm that this difference is due to glycolysis, we performed experiments where we treated the tumor tissue with glucose for 30 minutes and then placed it on a sensor. So the sensor response increases, the current increases. And in another sample, it was not treated with glucose, it was put in buffer solution for 30 minutes. So this is the response of that sensor. It shows a very low response. But after 30 minutes, what we did is we introduced glucose solution in the sensor and waited for some time. So after 20-25 minutes, what we observe is that the current increases and then it goes higher, somewhat similar to the tumor tissue. So this is because after glucose is introduced, glycolysis is set in, and then there's an increase in the response. So this is the response of the sensor for melanoma, mice melanoma tissue. So we've also validated the sensor using human tissue samples, breast tissue samples. We have a MOU with Tata Memorial Hospital, from where we get these breast tissue samples. So this is a response of human breast tissue sample for the cancerous sample and the normal. As you can see, the cancerous sample shows an increase in the current as compared to normal. And this is a response of 3 different samples which are cut from the same chunk of the tumor. So this is for the normal. Now, this is a response of the sensor for a sample which had a tumor content of 90%. Now this content is determined by IHC, and this is a response for 80% tumor content. And even for as low as 2 to 4% tumor content, we could get a clear-cut signal of increasing current, which could differentiate the cancer sample from the normal sample. So this technology for this device is available on the BRC website for private companies to take it up for commercialization. In fact, this technology has been transferred to 4 different companies till now, and they are working on the commercialization of this device. So to summarize, today I presented some examples of electrochemical and optical biosensor, which were used for detection of metabolite, where the enzyme substrate type of biochemical reactions were exploited. Some examples for cancer biomarker detection, exploiting the antigen-antibody type of interaction, and cancer detection in tissue samples by exploiting the difference in the glycolysis rate. So I would like to acknowledge my collaborators from Molecular Biology Division for the work on genetically modified bacteria, and my collaborator from Dr. Jaikumar, from IIT Madras, Professor Doble, Professor Srivastav, Dr. Shalaka Joshi from TMH, my colleagues Ms. Pallavi and Dr. C.A. Amarnath. Thank you.
Dr. Banji? Oh, yeah, great.
Managed? Can everybody see my screen now?
Yes.
Yes?
Yes.
Yeah. This is great. I think it's easier to do robotic surgery than... Okay, so everybody can see my screen, so that's good. And everybody can see me and hear me? Let me just apologize for the little confusion that happened. And as I said, these softwares can really confuse you, and I'm really hoping for some physical meetings now. But I heard very, very attentively to Anuradha, Geeta, and Dr. Pallavi. Excellent contributions to science. But I think it is kind of serendipity that I'm coming in the end. Because all the help that I can get with these 3 areas, that means the pathology, the diagnosis, and of course, the stem cell research. And I think in the end, it's my job to come and do a little bit of surgical work in the end. So I can see, but I'm not sure how I can start presenting my slides.
Keep moving. Just keep moving the slides.
Okay. It says stop presenting, actually. Why should it say stop presenting?
No, don't stop.
Ignore. Yes. Got it. There's too many things here, Okay. All right. Okay. So actually what you're seeing, what you're seeing actually is the PowerPoint presentation of my slide and not the slide. I don't know why the slide did come. And how do I move my PowerPoint presentation? I think I am in a little bit of...
could scroll down, keep scrolling.
Yes, that's what I'm trying. It doesn't scroll also. I was very happy that I could do something.
Maybe it takes few seconds, so... Possible.
Okay, so I am anyway prepared to even talk to you guys without any slides. That's not really necessary. So surgery is, if you look at surgery, in a nutshell, I'd only say one line. It's actually an assault to somebody's body. It's actually a crime if what we do as surgeons, if we don't take your consent before we do what we do. So it is that really invasive to anybody's body, and nobody wants to have a surgery today. So how are we changing the future of surgery by just doing something like what we call as robotics? I'm sorry, it doesn't move. Nothing moves here. I'm a little hard pressed here. What do I do? Stop presenting. Try again. Okay, let me share this. Is it better? Am I with you guys?
Yes.
Yeah.
Yeah, okay. Everybody can see my slides?
Yeah. Yes.
Okay, great. So that's what I was talking. So it's an assault if it is done without consent. Although the surgery has been described at Indus Valley Civilization many, many years, almost 2500 BC. And in India, we have a strong thing of surgery right from 6th century BC on the ghats of river Ganges, and that's Varanasi. I'd like to start talking to you regarding the future of surgery and robotic surgery by going back, as somebody was mentioning that we need to look back to be able to go forward, and that's your logo, isn't it? So I have actually been trained at Institute of Medical Sciences, Banaras Hindu University, on the banks of river Ganges, and my first encounter was this. So this surgery was an open surgery, which was happening in the department that I was working. And I realized that the average time to recover from this kind of open surgery was 6 to 8 weeks. It required prolonged immobilization, bed rest, pain and agony on these women. And of course, they could not go back to work, even look after their children. And this was the kind of incisions we were talking about. We were saving lives, yes, but were we giving them the quality of life? And that was the big question that was coming to everybody's mind. So how do we make this experience patient-friendly? How would you like to be absolutely comfortable at the thought of somebody advising you surgery? So just out of curiosity, let me understand how many of you actually in the audience have had a gynecological surgery or know somebody in your family who had a gynecological surgery. So if you have or had, you will realize that the keyhole surgeries, which started with the era of minimally invasive surgery, was more patient-friendly. It gave women to stand up on their feet faster with better surgical field and visibility in telescopes. So I'm audible, right?
You're audible, but the slides are not moving. Can you just try to scroll the slides?
It is not moving.
So go to the left. You have a panel. Yeah, that's perfect. It's moving now.
It's moving now?
Yes, perfect.
Okay, so we were just talking about laparoscopy, and we need to understand that this is a standard of care in terms of minimal access surgery today. And I'm sure all of you know about laparoscopy. That's how our theater looks. And that's how we perform laparoscopy, standing next to the patient, using straight stick instruments and going ahead with the surgery. We need to understand two gentlemen in our ability to do this minimal access surgery. One is Dr. Karma Nizar, who actually introduced the idea of fitting a video camera, putting inside the patient's abdomen to see the things inside and then operate. So that gave us some idea. And Dr. Harry Risch, who did the first hysterectomy, that means removal of uterus using the keyhole methods. But was it the panacea? It wasn't. And that is why the scientists and the surgeons and the physicians started looking for something more. There were challenges, and the challenges were basically 4. Now, if you can see this OT setup and the way we are operating, these movements are counterintuitive. That means they don't move the way my body moves. I'm unsteady, I have a 2D vision, and I'm limited with the instrument's motion, which is straight stick. And of course, if just imagine you have to do something at the end of a long stick, there will be a little tremor. And that was the problem. We also had a long learning curve. We had to convert the patients into an open surgery while doing. There was skill and ability of the assistant, which we had to depend on, and so forth. And that is why, instead of trying to do a tandav, which Shiv does, that means standing next to the patient, using both hands, using both feet in different positions and things, we wanted to actually start operating like Durgamma. We wanted to be absolutely cool, stable, multiple hands, and sit down and do our surgery. And that is why there was a change in the paradigm of surgery. From a surgeon-centric, it became patient-centric. We don't want to be doctors of this nature anymore, that they say, when you want your opinion, we'll give you one. You may come across some of them now, but no. Today, we want to do what the patient wants and what gives the patient comfort. So the concept of computer-assisted or robotic surgery came into picture. To the audience, again, I would like to ask, How many of you are using this phone? Probably none. All of you have moved to smartphones. And what is the reason? The reason is that a smartphone gives you much more technological advantage. And that's Dr. Frederick Moll, who was a surgeon in the US and started thinking of how the computer or AI or robot can help me perform my surgery. He is so-called the father of robotic surgery and started the intuitive surgical, the robots which we use today. And that's Dr. Mani Menon, who translated the idea of Dr. Moll into using it for radical prostatectomy and popularized it method so that other departments started using, and we as gynecologists were one of them. These are the timeline of innovations that the intuitive surgicals have made on their surgical robots. And today we are using the XI or the Da Vinci XI, the most latest robot at our institute in Apollo hospitals. So this robotic technology is nothing like this Chitty or the Ravan or the Terminator or any other robot you might have seen in the picture. It's actually a slave robot. It doesn't do anything other than what I want it to do. So it is completely under my control. It looks like science fiction. It has 3 parts. It has a surgeon's console. It has a patient cart, which has the robotic arms, and it has a vision cart. This is the actual pictures of these robots in our theater, and this is how it moves when we want it to.
Slide movement, please.
No moving, not moving?
No.
Can you see now?
We are at challenges in laparoscopy.
Still?
Yeah.
Oh my God. Okay. All right. Can you see a slide now?
It's challenges.
Challenging?
Yeah. Yes. Next one. Yeah.
Okay.
Yeah. You can see the.
You can see the robot?
Yeah.
All right. Okay. But it's not moving in this format when I'm talking. I can't move it in this format.
Is it? Can you make it into slideshow and then try?
Okay.
The bottom.
So I think bottom. No, this is not. All right. Okay. Slideshow. It will come here.
Yeah. Try now.
I think my presentation is too heavy for Google to handle. The Zoom handles better. Sorry about it.
Okay. Once. Yeah.
Now where did it go?
It's still there. Yeah.
I'm sorry, you guys. Sorry. Oh, I'm sorry. Anyhow, so this is it okay? Or we are gone into some other mode?
It's 2 screens, so.
I don't know.
You can open your presentation now.
Yeah. Can you see?
Yes.
Okay, so can you see the video?
No.
No?
No.
Can you see the robot?
Yes.
Okay, right, no problem. I think.
Ma'am, this is sufficient. You please go on. Yeah,
I will. So this is just trying to show you how the robot is used in the theater to get onto the patient for us to work on. Is the slide moving now?
Not yet. Yes. Yeah.
Okay, can you see a small instrument there on my hands?
No, that's the next one, I think.
Is the slide visible now?
It's one shot of the hand.
Okay. I think there is a lot of challenge here. No.
Yeah, it's now. Yeah.
It's better now?
Yes.
Okay.
Yeah.
Okay, so I'll actually show it in this format rather than making it full.
Yeah, now we see the video.
Is better? Okay.
You're seeing the video now.
Excellent. So you can see how the hands are moving to operate the robot?
Yes.
Perfect. So what I'm trying to make you understand that there are basically 2 parts of this machine or this tool that we call it. One is the one that gets latched onto the patient through which we insert those small instruments. And this is the console. You can see me sitting on the console and moving my hands, which is actually moving the masters, as we call it. And these masters, along with these foot controls, will help me perform the surgery. This is the operating room setup as it looks. There is a small video of a surgery. Is that visible to you? Am I with everybody?
Yes, it is visible, ma'am.
Yes, ma'am. Please go ahead.
Okay, so this is a small video just trying to show you. This is a video of a surgery that is going on, which we call as myomectomy, which we have done some time back. And this is usually done to remove a tumor from the uterus of a woman who is unable to conceive or is having symptoms of bleeding or pain. Now, this kind of tumor, otherwise we would have done an open surgery to remove this, but the whole technology helps us to do it with minimal access surgery, which is hardly not even 1 centimeter holes in your tummy, maybe 2 or maximum 3. And that gives a benefit. We had one patient whom we just operated yesterday. We did the surgery, evening the patient is back home, did not even needed to stay the overnight. So that's what we call is a patient friendly. But don't misunderstand me when I say everything is being only patient friendly. It's also surgeon friendly. It gives me a 3-dimensional, stereoscopic, immersive vision. And vision is everything, you can only do as much as you can see. The second is you can see these instruments moving. They have a wrist-like movement, which no other technology gives you. So I put tiny instruments inside the abdomen and they move like my wrist. They just follow what I'm doing on the console with my masters, exactly the same thing. They don't move on their own. They stop when I stop, and they help me perform the same task through a very, very tiny hole in your tummy. It also gives me additional benefits, which I'll just talk to you about. But what are the barriers in accepting it? Now, you'll not find not many surgeons talking about robotic surgery or even adapting in their practice. It's psychological. It's acquiring new skills. And of course, it's always economics. You must understand that we all are creatures of our habit. We want to resistant to change. New technologies are always disruptive. I'll just take an example of Uber. Now, when the Uber came, I think all the regular taxi fellows were worried that they will be out of work. But for a patient, for a person like me, it's so good. I just press a button and I know the car appears from somewhere and I go somewhere. But ultimately, the regular taxi guys also realize that it's a good way to move around because they get the customers on a tap. They don't have to keep on waiting in some corner and trying to wait for customers to come. So we must remember to embrace change where it can be. The second reason for not adopting such technologies is the fear of unknown. We don't know how it's going to work, how it looks. This is too big for me, whether it's approved, whether it's safe, whether I'll be ridiculed by my other colleagues. Well, we must remember that technology, and I think this is the right forum for me to talk, any scientific technology has its run. We must realize and use it to make sure that it does work and we will be able to use it for our clinical benefit. So don't be disappointed if nobody stands with you because you could many times do things alone. And also I would say that yes, looking back is important because the dots join in the back, and then you have to have the kind of trust either on your gut, on your destiny, on your life, or your something, that what my dots are actually joining in the back will also further join in future. So for me, this luck, destiny, opportunity started sometime in August 2012 when I was trained as a hardcore laparoscopic surgeon, and I thought I could do everything by that. But this was an advanced technology in supplementing or complementing my laparoscopic skills, and when I got introduced to the Da Vinci robotic system. And there has been no turning back, and I have been able to actually use this in so many situations where I would have felt inadequate using only laparoscopic work. So life actually always begins at the end of your comfort zone, and that's what we must remember. There are always criticisms. There will always be thoughts that one cannot progress or one cannot take this technology forward because it looks science fiction. And this is the same thing that happened many, many years ago, maybe 3 decades ago, when laparoscopy came into clinical use, and there were these questions that came up in an editorial. But it was interesting to note that the same gentleman, Dr. Roy Pitkin, 18 years wrote another editorial in the American Journal and said, no, in last 2 decades, laparoscopic surgery has got established scientific base, developed educational methods, and is an integral part of gynecological surgery. So today, we need to have patience. We need to understand that this new technology of robotic surgery, which I would say is not new to me because it's been 10 years since I'm working on it, and I would find that this is what happens. I agree with Mahatma Gandhi's quote that first they ignore you, then they laugh at you, and then they fight you. And then you win in the end. So today, robotic surgery is spearheading in all areas, and especially in benign gynecology, because I feel women who suffer from benign gynecological disease do not deserve an open surgery. They deserve the best of technology to be up and about on their feet. What extra do we have other than this? we have something what we call as a Firefly technology. Now we can inject a fluorescent dye into a patient and use the infrared camera on the robotic system and find out exactly where the disease is. This is a small clip of me doing an endometriosis surgery. Sometimes it may be difficult to find small lesions. So we use the Firefly technology here and exactly locate those lesions and make sure all of them are excised. So the normal tissue is saved, but the abnormal tissues are excised. And that's where the AI also is working towards helping surgeons identify normal to abnormal tissue. Virtual reality is almost knocking at my door. And I think by the time maybe I'll meet you people in a year or two, we will be showing you how we are using virtual reality in our day-to-day clinical management. We also are using 3D printing technology today to improve our work. Now, this is one paper that I just published in the Journal of Minimal Access Invasive Journal from AAGL USA, where we used a 3D printing model to remove all 12 fibroids in a patient's uterus who was young, unmarried, and obviously wanted to save her uterus for future pregnancy. And this is the correlation of the 3D model, which we kept in the theater while we did the robotic surgery and making sure that we did not miss out any of these tumors back in this woman. Today, the AI is actually moving very, very fast. We are almost at the threshold, as I said, and India is not far behind. We have virtual reality things in our lab right now, which we just tested 2 days back, and I'm sure I'll be using it in the clinical scenario very soon. Just trying to give you an idea of how surgeons will be using artificial technology, because we can recreate your pathology, anatomy, and exactly locate the tumors where they are. This is just a small video to show how they are locating a small tumor in the upper lobe of the lung. We're using the robotic, the AI, and the 3D PDF format to exactly understand the patient's problem, to locate it where it is. Once it is located, then you prep up your theater, take the exact points, take your team, and which is very important, and go and remove the tumor using the modern technology. So these are some things that can be very useful. But not to forget, this technology is also to be used to train our future surgeons. Gone are the days where surgeons will see one and do one. It's no longer there. Would you sit in an aircraft if the pilot says, I've seen somebody flying an aircraft and I'll take you? Nobody wants to do that. The surgery also should not be the same. There is also a plethora of these robotics platforms that are coming up. This is a Teleflex, which is a European company. This is a CMR, which is also being used in India, a company from UK. This is Revo from South Korea, and that is Hugo, a Medtronic company. So 4 or 5 robots are, other than Da Vinci, very, very close to using clinical practice.
You have 2 more minutes, Roma.
Ma'am, I'm already done. So I would take any questions if there are. We love the work that we are doing here in India, and proud to say that I don't think at any stage we are anyway far behind of what anybody in the world is doing today. So technology is borderless, and we get the same technology what anybody else in the country does. But yes, talking about my surgical team, I mentioned you cannot perform anything in the surgery without your surgical team, but not to forget the team at home. Without their support, we will probably not be able to do what we are able to do today. So thank you very much for the patient hearing. I would take any questions if I have 2 minutes. Otherwise, I'll stop my presentation.
Thank you very much, Ruma. I could see the amount of effort that has gone in preparing these slides. I'm sorry for all the trouble you had.
It was
now I leave it to Geeta to decide what to do. We have cut into our tea time, but we do need a break. So Geeta, take over. I'm leaving.
1 or 2 questions are welcome. So maybe I already asked, and Shilpa has already responded to mine.
All the questions can be put in the chat box. Yes. Questions can be put in the chat box, and they can be answered by the speakers virtually.
Yeah, we already started. Because Ruma, just one question.
Anyway, I leave the decision to you. You are the organizer.
From laparoscopy, we can go to open surgeries. So from robotic, what is your backup plan if something goes wrong, and what are the limitations?
Okay, so just like today's situation, it's always fluid. That's why the surgeon always has to think out of the box situations where it can be in some kind of trouble, and we can be sometimes. Any surgery can be converted to open surgery. So that's always a backup plan. And that is what we teach our students, that you first master the open surgery, because that is always the backup plan. And the steps are the same. There is always an emergency undocking procedure in the OT. We take 2 or 3 minutes to undock the robot and proceed with whatever we want, if it's an emergency. If it's not an emergency, you can take your time and still convert. So when we have to convert from minimal access to open, we talk about two things: strategic conversion and emergency conversion. So sometimes we take a strategic decision that yes, it is safety from the patient's point if we don't do it, continue with the minimal access, but do an open surgery. So that's a strategic decision. But sometimes it can be an emergency. So we have a method of doing emergency undocking and proceeding.
Wonderful. Yeah. Any questions for Shilpa? She's already answered mine.
Just to share, Geeta, my nephew does robotic surgery in nephrology in US though.
Perfect. So nice.
So when he started, we had tons and tons of questions to ask him. So
very nice.
That's great.
Yeah. Thank you.
Any questions for Anuradha? Yeah, okay. So if not, so since we are 20 minutes late, so is it okay with all of you if we go ahead? You can continue to sip your tea and take bio breaks.
Yeah, I would say yes to this.
Okay.
Because paucity of time will add up in the next session.
Yeah, I don't want to extend the last session. That's the reason. So thank you so much for your cooperation. I would, so if the questions can continue to come in the chat box or in the email, I'll forward it to the speakers. And on behalf we have to check the weight of the presentation before we use the tool. So heavy, heavy presentations probably don't go well with some of the software. So that's another learning curve for us. So we would move on to the second session. And it's my pleasure to invite the chairpersons and the moderators. The chairpersons, Dr. Jyoti. I would introduce Dr. Jyoti, and then she would introduce the moderators. I have known Jyoti personally, and when she was heading the Institute of Genetics, and I remember those years were where we used to have most regularly DBT workshops. Practically every year we would keep a slot free for her. So such is her academic interest in holding workshops. She has a very rich biodata. She works in medical genetics, cytogenetics, has guided more than 15 students and more than 250 projects. She's a recipient of many awards, more than 182 publications, and she is a member of many professional bodies. Overall, a wonderful human being. Therefore, that's why she's wanted in all the committees. So with that brief introduction, Dr. Jyoti, I request you to chair the session. So you can introduce the moderators, and then you can invite the speakers one by one. Thank you so much. Please unmute yourself, Dr. Jyoti. The co-host.
Dr. Jyoti, you're not audible.
Yes. Dr. Jyoti, please unmute yourself.
We can't hear you, Jyoti. We can't hear you.
Just give us a minute. You are the co-host.
Yes, we can't hear Jyoti.
Can you...
Thank you.
We can't hear you, Jyoti.
Okay. Can you hear me now? Hello?
Yes, we can hear you. Yes.
Okay. Okay. Thank you for your kind introduction, Dr. Geeta. Very good afternoon to you all. Friends, it's a proud day for all of us to celebrate the Golden Jubilee year of the association, beginning with this notable webinar focusing very aptly on the subject Advances in Technology in Health Sector. I congratulate the organizers for taking this great initiative and hosting this historic event. Now I welcome you all to the second session of the webinar. This is a 2-hour session with 5 distinguished speakers. The talk time for each speaker is 20 minutes, and we have question and answer section at the end of the talks for about 15 minutes. Now, may I now we request all the delegates to post your questions in the chat box. For leading this session, I now take the pleasure of introducing the moderator, a renowned scientist, Dr. Kaiser Jameel. Dr. Kaiser Jameel is known to many of us, and she is presently the head department of genetics at Mahavir Hospital and Research Center. And she has 4 decades of research career and has guided 45 research students for project work. She has published more than 300 papers in national and international journals of repute. She has several awards and honors for her credit, and she's a life member of several bodies, and she's a life member of this IWSA. She's also the first woman from India to be elected as president of Organization of Women in Science for the Developing World, with its headquarters at Trieste, Italy, which she served from 2005 to 10 with the national chapter of OWSD for Indian women scientists, where she's the president now. And she is engaged in cancer biology projects, and she's an editorial member of many prestigious journals and reviewer of many journals. And her passion is human dimensions, a vision for sharing, learning, and transforming. Over to Dr. Kaiser Jameel, ma'am.
Thank you, Dr. Jyoti, for the kind introduction. So let us get the ball rolling by introducing the first speaker of this session, who is Dr. P Manjushri. She's a scientist, GSG, and head Flood Modeling Division, Disaster Management Support Group of NRSC, ISRO, Government of India organization. So she is a accomplished scientist, and she completed her BSc and MSc from Osmania University and did her PhD from JNTU in 2016 on the topic of spatial information technology. And she taught in colleges for 2 years, and she's currently heading the flood modeling division at ISRO. Her research achievements, she has specialized in geospatial technologies like remote sensing, GIS, web technology for hydrometeorological disasters, and disaster risk reduction at various levels of government machinery. And she's responsible for preparedness and release of the first spatial flood hazard atlas for Bihar state. So her contributions, she has more than 40 scientific publications in peer-reviewed international and national journals, and in seminars and books with 300 citations. She has mentored about 10 MTech and MS students for their thesis, and she received 3 national and international. She is a reviewer of 3 national and international journals. She has several awards and honors, and she has been conferred with the Distinguished Scientist Award in Remote Sensing in 2018 by VIRA, Chennai. ISRO Team Excellence Awards for ISRO DMS program in 2007 and 14, and ISRO Team Excellence Award for Bhuvan Geospatial Services in 2013. She received appreciation from central and state government departments and nodal ministries for her dedication and sincere efforts. She's life member of our IWSA, and she's holding executive body member in our IWSA. Over to you, Dr. Manjushri.
Thank you. Thank you very much, Kaiser ma'am. Good evening. Good evening to all. Am I audible? Yes.
Yeah.
Thank you. Thank you very much. So first, let me thank Professor Geeta ma'am for giving me this opportunity, and Ratna ma'am and all the members of IWSA for giving me this opportunity, and then presenting the space sector, how it is useful for the medical science and the medical sector. Before that, I just wish I don't get problems like Dr. Ruma. So I try to share my screen now. Is the presentation visible?
Presentation. Keep your presentation ready. Probably you click many times, so.
Presentation is not visible.
Open your presentation and then share.
Is it fine? Yes. Is it okay? Am I audible and the presentation is seen?
Yes.
Yeah. Okay. Also, the topic of the day is the space research for health. So I would like to start saying that a few verses from Sanskrit verses. Loka samasta sukhino bhavantu. All the world be happy. And this is possible only through Arogyame Mahabhagyam. So health is the biggest wealth. So from almost from 2 hours, we have been listening to the great work which is being done by the researchers, and also in terms of the technology, especially the medical technology, the advancements using AI and robotics, everything. Now I come to a different sector. This is the space sector. So as we all know, that ISRO builds vehicles, it launches vehicles, and then it will design the satellites and the sensors inside. And we have a host of Earth observation satellites, communication satellites, navigation satellites, and also the ground sensors and also the satellite communication devices which are used at the time of emergencies and also for other
Sorry to
things.
interrupt. Are you moving
And
your slides,
apart from
Manju?
this,
Are you
no,
moving your
not it. We are also moving. No, not it, ma'am. I'm in the first slide. So apart from that, we also have the planetary missions. So let me go a little bit in detail about the Earth observation satellites, which basically are responsible for providing information on land and water, infrastructure, ocean, weather and climate, and which has a capability of imaging the Earth from 1 kilometer resolution, spatial resolution, to 0.65 meters and less than. Now it is possible less than 0.5 also. So and there are continuity missions for all the existing which are going on. We also focus on the continuing missions of this because the data is very, very important for doing the research. So our EVO program consists of a constellation of the satellite resource sats, ocean sats, and cartosats. Now, how these are useful in our health sector? So the most important thing is they provide a wealth of information on the environmental factors that influence the mosquito-borne diseases, especially like malaria, dengue, and West Nile virus, which is very popular, and keep the human population at risk. So the long-term observation, as I mentioned, the continuity of missions help us in providing the long-term observations, and also it facilitates the analysis between the climate and the malaria relationships. So the high-resolution data can be used to assess the effects of agriculture, urbanization, deforestation, and water management on the malarial spread, on the larvae spread of the malaria. So since 2000, there is a considerable progress in reducing the global burden of malaria and shrinking the malaria map and moving towards the goal of malaria eradication. So the recent Lancet Commission report on malaria eradication emphasized on the geospatial data, which includes the satellite observations as a key data source and development of decision support tools to enable the health management teams and also the doctors, and so that a national and subnational program management at a regional and global strategic planning can be done for this. So we have here a host of satellites. There, I'm showing here the spatial resolution. There are so many satellites. These are not only Indian, but also includes the foreign missions, where we have the resolution ranging from, as I mentioned, from 1 meter to about 1
Manju, the
kilometer.
slides are not moving, sorry.
Yeah. Is this okay? So
no.
Is this fine?
We are still on the first slide.
Oh no. I think I'm getting the same problem as Dr.
Scroll down, because if you're on the slideshow, then your down movement will work. Otherwise, in the left box, you have that. Keep moving that slide.
Is this? Are you able to see my third slide?
Still on the first.
No, click on the third slide.
No, full screen is not there. Should that be a full screen?
Yeah, now it's moving. Satellite-derived parameters.
Yes. So I'll do one thing.
Keep full screen.
I'll present it like this. I think this is. So what do we get from a satellite? So what we get is the precipitation, the land surface temperature, and from this we also get the climate parameters, the temperature, humidity, precipitation, and also the spectral indices can be designed using these parameters, and we get the water bodies, the soil moisture, and the land use and land cover maps, the population density, and the building maps. So these are some of the parameters which help in allowing us to make models and also see where exactly the larvae or the malaria spread happens, and to combat different procedures, how to combat this spread of the larvae. So the types of variation because of the geographic patterns and the seasonal cycles and interannual variability and long-term trends will be there, and this will be go as an input to the model. So generally the mosquito habitats are the temporary water bodies, and the vegetation greenness we get, and vegetation type forest, grassland or barren. And coming to the land use, so the different types of settlements patterns are there, and they form the living condition, they design the living conditions and the activities on the human, and how the humans are exposed to the mosquitoes, and this is all, and how the mosquito parasites are reproducing, and where are they reproducing, what are the areas where they are getting breed. All are possible to find from the satellite data. There are various case studies. Here I have mentioned few case studies where a lot of studies have been made using the NASA satellite products, and where they have been incorporated, especially in into the US Armed Corps, where the spatial analytics for force, health, and readiness. This program is called the VAST to predict the occurrence of dengue outbreaks in Cambodia. So similarly for West Nile virus also, the forecasting and risk mapping is done by the South Dakota State University, where they have used web-based information systems to integrate the mosquito surveillance data with the environmental data which comes from the satellite. And the malaria early warning of Ethiopia. So these are the different types of products which have been generated, and they have predicted where the high risk villages are there due to different species of malaria also. Yeah, coming to the communication satellite series. So we have here the INSAT series, which is actually a very big fleet of satellites are there, which are basically used for the telecommunication, the telemedicine, education, and it provides the mobile satellite services, radio networking, the village resource center, and satellite aided search and rescue, and news gathering and dissemination, television, and also training and development communication programs. So this has got a wide area coverage, a reliable data delivery, and robustness and broadcast are the unique features of this satellite system. So the most important application here is the telemedicine, and which has been demonstrated and proved that through telemedicine how success cases are there, where you can demonstrate the success rate of the telemedicine technology. And it is possible both right now through satellite and cellular network, and they are also using for the various medical fields, including the dermatology, pediatrics, and surgery. And telesurgery has been tried on internal mammary artery dissection in pigs with robotic technology to determine the feasibility of the bandwidth requirements. And also telemedicine using the satellite communication is also useful for a country's defense system by equipping the Navy ships with the telemedicine kit. This is one of the most important strategic application of the telemedicine also. And medical teleeducation, the practice of new or continuing medical education via distance learning is also being done. Now, another class are the navigation constellation of satellites, where it provides the geospatial positioning of a person across the globe. So this consists of a constellation of satellites. About 7 satellites are there. All are in place. We have now our own Indian regional navigation system in place, and the standard position accuracy is better than 5 meters with an accuracy of 40 nanoseconds. So this has got a wide applications like the maritime services, geofencing, geoservices, etc. But the issue point here is, if we are using the GPS along with the GIS, which is nothing but a spatial system, then we can actually do a lot of application tools and application developments are there, which are very much useful for the medical health sector. So I'll be discussing some examples how we have used both this GIS and GPS. So in totality, the confluence of the satellite technology in the complete life cycle of the healthcare, which is nothing but the prevention, preparation, then the monitoring, management, and mitigation. So in all these 5 parameters, of the healthcare management, satellites play a very important, and they have their role and their footprint in providing different types of data sets. So let me just talk about the telemedicine. So just mere lack of disease is not health, but the well-being in both physical, mental, and social aspects is called the true health. So the healthcare involves prevention, maintenance, and also management. So telemedicine is useful in all these three phases as discussed in the earlier slide. So it has actually started somewhere in 2001. So ISRO was the first to demonstrate how telemedicine is possible through a satellite. So the evolution has taken place. Initially, it was from point to point, but whereas now it is from multi-point to multi-point. So this is how the treatment is being done remotely through the satellite technology. So a very successful application was during COVID-19, where the video visits were taken place in place of the personal, and also useful with testing till declared positive, and smaller subset of high risk patients only move from telemedicine to conventional health care. So non-COVID patients can receive care from home and distance, and reducing the burden on hospitals because of this, and minimizing the risk to the healthcare workers. So the paradigm shift is, the positive outcome of COVID is the technology industry push from one side, and the consumer patient pool is on the other side. So for almost 2 decades, this was a very successful, and the IRDA instructed all the existing insurance policies also to cover telemedicine as part of their policies. So the insurance companies also started to recognize the teleconsultations, which is a positive outcome of the telemedicine. So the advances driving them is, now there are a lot of advances in the technology, especially in the communication technology, new satellites and new bandwidths, and the electronics, new biosensors, as mentioned by the previous speakers, how the technology is improving, especially in the designing and development of the electronic biosensors, and the information technology also, a lot of things, and now it is the cloud computing which we are talking, and the medicine and application. Always, there is, as earlier speakers have mentioned, there is a lot of research going on, either in terms of surgery or in terms of the stem cells or whatever it is. So the medicine and application research. So this complete cycle, the propelling factors to have a new device, new things, is the convergence of all these technologies. So the recent advances, I think this is the miniature of electronics is now what is happening. So the handheld medical and diagnostics devices, and increasing penetration of mobiles, the power of mobiles in display and computing capacity, and the number of apps are increasing, and expanding reach of connectivity, and the option of operators also is increasing. The cloud computing options are also many now. So we have a lot of challenges with the advancement in technology. We also have challenges too, as earlier speaker Dr. Roma has mentioned. We always have a resistance towards anything new. So but once then people start adjusting to the new risks. So the challenges ahead are the changing patient physician needs, and coming out with a more user-friendly technology with easy interfaces, and the reinvestment costs also should be taken care, and the maintenance, interoperability, privacy and security of information, and user acceptance. So telehealth serves as a beacon of hope in the future of medicine. As we have seen, we have never expected a COVID as a corona as one which will actually affecting us for almost 2 years. So we need to be prepared. We need to prepare ourselves for any type of medical challenges which are coming in front of us. So no one knows how the corona is, how it is mutating and all that. So the forward year telemedicine was very successful. For these are some of the listed. I have listed some of the hospitals and also the medical institutions which are using, and of course the internet broadband is there, wireless state WAN network is there, and also the grids are also there, and many it is also being used for many diseases right now. And Shankar Netralaya and Arvind Eye hospitals are the ones which are successfully using the telemedicine technology right now. So the initiative is in March 25th, 2020. So the Ministry of Health and Family Welfare, Government of India, has recognized this as a new tool for national healthcare delivery system. So the guidelines given were the definition of the telemedicine and telehealth, and there were some certification courses and tools for telemedicine and the storage of records and equal healthcare access to all. All these things have taken place with this recognition, and now people are we are actually contemplating to have a telemedicine grid of India. So one country, one healthcare network, with about 160,000 health and wellness centers across India being connected. And in the phase one, about 60,000 will be connected. And many public networks like ISRO, Telemedicine Network, and NKN will be included in this. Many private hospitals and also household connectivity also in some states are being thought of. So that is with respect to the satellites, how they are useful in this sector. Now, one more important technology is the geospatial technology. This has been never thought of earlier, but now after the COVID, and now this has become a very useful tool, how the geospatial technology can be useful to tackle this type of diseases like the corona, epidemic which has happened. So what we have done is we have actually supported various state governments. ISRO has supported various state governments by providing the geospatial tools and also the location-based solutions through GPS to fight against them, including the national level coronavirus tracker. So the impact studies were done so that the atmospheric parameters and also the water themes can be delineated from the satellites. So the impact studies was done with these parameters, and these tools and services have been gainfully utilized by the state governments in the country. So this is the Bhuvan platform, it is called, and this shows how, if you just click, this is in all the geospatial way. The information is available on the Ministry of Health and Family Welfare website, and we have taken the information from them and then seen that how geospatially you can show the information so that it will help the medical health workers as well as the health management. So this shows how the state-wise COVID cases are available. And this is an example. If you go now and see this website, here you can see the vaccination centers around you, and also the age groups, the dosage. These are some of the things which you can actually enable and then try to know. This I have given it yesterday here in this area of Shadnagar. How many vaccination centers are available for this age, about 45, and also for dose 1 and dose 2. So this is the table which I have got. So it says that in which vaccination center, how many are available, and on which date, how many are available. So this sort of information is very easy to understand, and this is a time series information on the COVID-19 cases. This is how the monitoring and managing of all these cases is. It is very complex, but if you use the geospatial technology, it is possible to see at one go what is the information available to you on the portal. So this shows the COVID-19 statistics. This is yesterday I have accessed this, and this is how the present growth status is, where Kerala is having the maximum number of cases, and this is a pan-India scenario, and the deceased cases in each state-wise statistics are also available. So one more thing is Bihar is one of the state which has used, and we have customized for Bihar a specific tool for them to mapping the epicenter of the infection center and depict the containment zones and hotspots for containment with territory buffer zones also, so that they can manage the pandemic at a very field level. So this has implemented as per the government stipulations and to effectively fight, localize, and contain the virus. So at the same time, very high resolution satellite images are able to provide the household level information, and this has actually gone to those villages survey also was done with respect to the each household. In this way, the information has been gathered and then collected and then it was displayed. So this is one of the image which is showing that how many villages have been affected in different villages, how many villages are affected due to this COVID, and also with a buffer of about 10 kilometers and also 5 kilometers, what are the villages which need to be taken care. Now, one more thing is the patient information system through Bhuvan for geovisualization. So here, Monal 2020 is one of the IoT-based technology. It's a designed by ECIL. It consists of wearable instrument for monitoring the COVID-19 patients' vital parameters like body temperatures, blood oxygen level, respiration rate, heart rate, etc. So these were we have made a this enables the round-the-clock monitoring in case, especially in case of high home isolation of the COVID-19 patients. So we have extended this report to use the Bhuvan services for geolocation of these patients in different zones. So this has helped them to understand the complete picture of the patient record, the history for this. And this is how
summarize?
Yes.
yeah, sure.
Yes, 4 more
Now,
minutes.
this is a World Health Map, which shows the COVID-19 at a different. So, what I mean to say at the end is, if you have a geo map like this, and continuously information start coming up, and then we design tools to capture this information and record it in front of the health management, then it is very easy for them to manage which to address first. So in summary, I say that remote sensing brings new tools for monitoring diseases and investigation their association with the multiple sense parameters, especially the environmental parameters, and ultimately creating an intelligent alert system. So achieving high spatial and temporal resolution, either by the hardware improvements or various numerical models are there, and important challenge in the remote sensing area. So the synergy between the remote sensing, GIS and GPS is nothing but it leads to the global health, and also involve epidemiological methodologies for activity tracking. This is also important, and cloud computing platforms and combining the space, cyber and ground data holds a very great potential. So this has been, I have to thank Dr. Murthy Remella, who is a scientist, senior scientist at ISRO, and who is also the Honorable Secretary of the Telemedicine Society of India, and who has given me some information about the telemedicine. And these are some of the acknowledgements which I would like to thank, where I have taken some information from various. So way ahead is it's to leverage furthermore new powerful modeling approaches like AI and assembly modeling, and new sources of very high resolution in collaboration between the MBD and remote sensing communities, and research in outer space have been very active in developing telemedicine, which I think we'll be going to have a lecture in the last. And also numerical examples of space technology applications exist, but sufficient training, awareness, and collaboration with the research community is a need of the hour. So interdisciplinary collaboration and an easier access for health researchers to space technologies is required. So this is what I would like to share. So thank you very much for your kind attention.
Thank you, Dr. Manjushri, for that excellent presentation, giving the importance of geospatial tools for the detection of so many COVID cases and the centers and so on. It is very, very useful. But we'll go to the next speaker. Professor Priya Abraham. She will be speaking on multidisciplinary approach to the pandemic. Professor Priya Abraham is the Director and Scientist G at the National Institute of Virology, Pune. Soon after joining as professor, Abraham found herself leading ICMR and IV, which had one of the most pivotal roles in India during this pandemic. Garnering the skills of a scientist, she and the team were able to report many firsts in the India. First cases of SARS-CoV-2, first electron micrograph, first whole genome sequences, first virus isolation, first indigenous ELISA, the first indigenous vaccine in partnership with BBIL, and the report of the first variant of concern in India as well as the subsequent variants. She also received the Business Excellence and Innovative Best Practices Academia Award in 2020 from the New Delhi Institute of Management, and the Janani Award for Medical Research Leadership, and the Rotary Vocational Excellence Award, Dharwad, Sharada Shakti Award, and the J.C. Bose Award. Prior to joining ICMR, NIV, Professor Abraham worked in Department of Clinical Virology at Christian Medical College, where she chaired the department earlier. She has been funded by DBT, ICMR, and NIH for her work in human papillomaviruses. She has won 76 publications to her credit. Now over to Dr. Priya Abraham.
Thank you very much for your kind introduction. I'm just trying to get my presentation on board. Just a second. Having some glitches here. While I'm having that sorted out, I want to convey my sincere thanks to the to Dr. Geeta for inviting me to this talk, and the chairperson, Dr. Jyoti, moderator, Dr. Jameel, and it is truly a pleasure to be amidst so many accomplished, eminent women scientists. And to see the kind of contributions these women scientists that I've heard have made, both not just in science but in healthcare, and also using remote sensing approaches, has been truly an eye-opener. And with that, I will just begin my talk. I hope you can see my slides in full presentation mode. Yes. And am I audible?
Yes.
Yeah. Okay. Thank you very much. So this was the topic given to me, a multidisciplinary approach to the pandemic. So I will speak a little about India's approach to this pandemic, touch upon few areas. ICMR NIV, the National Institute of Virology NIV, is part of a larger umbrella organization known as the Indian Council of Medical Research. So NIV's contribution and also how it intermeshes with our umbrella organization, the ICMR. So with that, let me continue. So we know that this pandemic, despite our huge advances in science, had really brought all nations to their knees, even the most advanced nations. Several facets of our existence took a very deep hit, be it economy, healthcare, education, safety and security, climate, and even because of the immense amount of misinformation and disinformation, there was a reduced trust in governments. The low and middle income countries particularly were badly affected because of the huge challenges and vulnerabilities that are prevalent in these countries. And in India, in addition to these challenges and vulnerabilities, its huge population and disparities definitely posed significant challenges. Just to touch upon some of India's responses, we know India started out even just past the middle of January with thermal screening of incoming passengers from other countries. There were travel advisories that were issued. India understood soon that it had to expand its testing capacity massively, and that it did so over time. We set up a call center. There was expanded a telemedicine e-Sanjeevani from the government, though many other telemedicine portals have been opened in other institutes, even in private institutes. But this e-Sanjeevani was able to access individuals in remote and hard to reach areas. The launching of the Arogya Setu app, then with a drive towards self-sufficiency or the Atmanirbhar initiative, many initiatives were taken, became self-sufficient with personal protective equipment, self-sufficient with our, ICU ventilators and other equipment, eventually even with oxygen plants, and then of course with our own testing capacity. Self-sufficiency soon was expanded to the manufacture and delivery of vaccines within our own country. So we were not as badly hit as some of the nations in Africa, for instance. We are an constitution of a national expert group on vaccine administration for COVID-19. We all are familiar with the CoWIN app, and we have at least 3 vaccines that are in use in our country, including vaccines for children aged 15 to 18. And we definitely have at least 3 more vaccines in the pipeline. So with that introduction, and in fact, where I had in my earlier slide shown the asterisk, it denotes areas where the National Institute of Virology was allowed to make some inroads in taking the nation forward. So these are 2 images of our 2 campuses of the National Institute of Virology, and here are our contributions. So very early as Jan 10th of 2020, physician scientists in my institute as well as laboratory scientists got together. Physician scientists were looking carefully at this pneumonia of unknown etiology that was being reported in Wuhan in China. They geared up with a medical response by making these sample case report forms, specimen referral forms, etc., modeling that from the WHO document. And laboratory scientists started doing some wet runs with the reagents they have to see whether they could pick up coronavirus from cases that presented with severe acute respiratory illness. And lo and behold, by Jan 30th, literally about 20 days from the first time we deliberated, our institute was the first to report the first 3 cases of SARS-CoV-2 in our country. And because of our wet runs, we were able to put together reagents, standard operating procedures, send it to certain key government labs known as the Virus Research Diagnostic Labs. These are part of the Department of Health Research and ICMR network. By the first week of Feb, we had sent out reagents to geographically critically positioned labs that were close to major airports. We sent out in-house positive controls to them, and capacity building grew massively till we had at least 107 of these VRDL labs using our reagents for an RT-PCR, and also many of the government labs. We became a kind of factory outlet. You can see we had standardized this PCR on our own. We put together the reagents, did a QC check, and these are multiple boxes lined up in our garage. And we dunked in dry ice, threw in the reagents, and these left, they were packaged, and these left our institute, be it by the last flight out of Pune or the first flight out of Pune, sending out, as of date, 64 lakh RT-PCR reactions to different government laboratories in the length and breadth of the country. We, in addition to this kit supply, this miniaturized kit that we put together, we also went about quality control, and along with our ICMR headquarters, we embarked on a quality control and quality assurance program where we used WHO SARS-CoV-2 quality assurance panels that we allocated and sent to over 1,300 government and private labs, because it was extremely important that not only should we send out reagents, but everybody should be putting out quality results. We also have filed some 9 patents over the last couple of years. Most notable are number 1 and actually number 7 and 6, which are actually companion patents, where we put in a multiplex single tube PCR assay, and we also put in RT lamp assay. 6 and 7 go together, where this, as you know, a lamp assay is a much more easier assay to do. It's isothermal, it has simpler instrumentation, it is quicker and cheaper. So one of our scientists, Dr. Nandi, and his team put together this, and this has been filed for a patent. The first electron micrograph picture we put out literally 2 weeks after we detected the first cases. This is the EM picture. And we also put out our first 2 SARS-CoV-2 whole genome sequences, which spanned 30,000 nucleotides of the virus. And we could see clearly, even at that time, amongst the first 2 strains that came into our country, they were genomically little diverse. You can see the arrows pointing to different points in this phylogenetic tree, these two being our first two sequences. We also were very lucky in isolating this virus in March 9th of 2020, and we interrogated using immunofluorescence, again, immune electron microscopy, and doing a whole genome sequence. We've also called to do kit validations of many kits that came to us for approval. At that time, by April, May, the whole world was running out of kits, and it was very important we identified a whole slew of kits so that we could continue our testing. This could be in the form of virus transport medium, RNA extraction kits, or RT-PCR kits, and so many were sent to us, of which only a proportion of them were found to be satisfactory, including some made in India kits. This was a powerful, leg up for the Atmanirbhar Bharat initiative. More recently, we have been validating kits that specifically detect the most recent variant of concern of SARS-CoV-2, namely Omicron, and we could actually approve only 2 out of the 12 kits that came to us. Another challenging mission that we were put to was that in March, I was informed by the Director General of ICMR to send out a team to Iran, where there were many cases of SARS-CoV-2 and there were several deaths. Several of our Indian citizens, students, and pilgrims were stranded there. So we sent out a team of 6 who collected samples from over 2,000 individuals and packaged it back to India. So they sent out around 2,028 samples, which were sent back to NIV Pune, and literally over the night, we screened them, and we were able to screen out those who were uninfected, and they could be quickly repatriated into our country. This was possible because of the exceptional cooperation from the diplomatic interventions by the Embassy of India, as well as in Iran, as well as the close coordination between ICMR, Ministry of Health and Family Welfare, the External Affairs Ministry, and the Indian Air Force and private airlines. We also were asked to be part of many training sessions. We trained the NDRF, many medical colleges in Maharashtra. At that point, early in the pandemic, we only had about 13 training sessions. Now, of course, we are back to physical, face-to-face trainings. But at that time in 2020, we had only trained face-to-face about 155 individuals. But online training were literally too numerous for me to mention. Literally 2 sessions, 1 in the morning, 1 in the evening, every day, 7 days a week. We also were able to make India's first indigenous ELISA kit, which was an IgG antibody detection kit against whole virion antigen of the SARS-CoV-2 virus. This was used for screening both in the clinical trials as well as the first sero surveys done for the nation. We were also asked to screen several compounds. Some of them were very strange compounds, to look for their antiviral activity against SARS-CoV-2. We interrogated over 100 compounds, of which we could only find only 22 to be satisfactory. Another request that we encountered, this was when convalescent plasma therapy was very much in vogue, and it was difficult to get enough convalescent plasma. Companies approached us whether we could give them the inactivated virus antigen so that they could make equine hyperimmune gamma globulin, which is akin to the anti-snake venom that we would issue if somebody had a snake bite. So they could make large amounts of this hyperimmune globulin, which would double up like convalescent plasma that was being used earlier on in clinic in therapy. So there was a lot of translational research that happened at that point in time. We helped a particular company make a point-of-care assay. We also transferred the technology of the whole virion assay, which we had called the Kavach ELISA, indigenous ELISA, to 7 companies, which was part of the initial Sero survey. And of course, the convalescent plasma trial, the Placid trial, the Platina trial in Maharashtra didn't really take off. And then, of course, companies that came to us for antigen, particularly to make hyperimmune equine gamma globulin. We did antiviral testing, and then, of course, our initiatives towards helping a pharma with vaccine. I'll come to that. We've also looked at the presence of the virus in sewage water, because that would be an indicator of the amount of SARS-CoV-2 cases in a particular region. And we were able to rope in 9 different labs across the country, and our results suggested that the SARS-CoV-2 in sewage water correlated well with the trend of SARS-CoV-2 19 cases in Mumbai, which was later found to be also correlating in the other 8 sites. So these are images of how our technologists sampled sewage water, be it from manholes or from sumps. They took the samples and then tested them using RT-PCR to see whether it mirrored the number of cases in a particular geographical region. We also asked to become part of a big biorepository network. So we are now holding several clinical specimens from cases from individuals who were SARS-CoV-2 positive. We have it here in Pune, as well as 2 of our field units in Bengaluru and Kerala. Then came our initiatives towards the vaccine. And you may recall I said that we isolated for the first time the virus in March 2020. And actually, literally a week later, BBIL approached us for wanting to partner with us to make a whole virion inactivated virus, a vaccine candidate. We further characterized the strain that we wanted to give them, gave to them at end April. They worked on the vaccine, on the virus, inactivated it, gave it to us for cross-checking, and then began our preclinical studies in hamsters and rhesus macaques in June to August of 2020. And by July, August, having got success with our preclinical trials in golden hamsters and rhesus macaques, by July, August, began the phase 1, phase 2 clinical trials, and then on to the phase 3 clinical trials. So
2 more minutes.
Sorry?
Professor Priya, 2
Yes.
more minutes.
How many more minutes?
2 more minutes.
Okay, so these are studies in hamsters. These are studies in non-human primates, which we did. And this is, of course, the much awaited phase 3 clinical trials showing the efficacy. And these are overviews of all the isolations of different variants, the alpha, beta, gamma, delta, what have you, and even Omicron. We've just got news that it's been accepted in viruses. And we've also been part of the whole genome sequencing consortium of India. Here is NIV along with 9 other central laboratories. This network has expanded to now 52 labs. And we have done several studies interrogating the antibody response in recipients of Covaxin, Covishield, and Sputnik to see whether there's antibody escape to the different variants. So these are studies basically of that. We also looked at individuals who had got heterologous prime boost, meaning first they got Covishield and then Covaxin accidentally to see whether the immune response was better. And indeed, we found that it was better if they got a heterologous prime boost. We also interrogated those who had breakthrough infections in our country during the second wave, and we found that 83.6% was all due to the Delta variant. And these are ICMR efforts in the companion efforts. We have several portals that tell you how many tests have been done so far, how many in a day, where you can access specimen referral forms, how to do sample correction, storage, and transport, kit validation portal where we feed it to, and reagent supply, telling us where all the kits are stored in different geographical regions, an inventory portal Interlab laboratory quality control portal, testing and stock status portal, vaccine portal, national clinical registry portal, a novel claim portal where people claim novel agents, novel kits, novel devices. And what more going forward? We know today the government has launched the Pradhan Mantri Ayushman Bharat Health Infrastructure Mission, which not only looks at expanding infrastructure for health and wellness centers, looking into primary healthcare, but also bolstering secondary and tertiary healthcare, increasing the community engagement, but also improving disease surveillance to support generating evidence and forecasting potential outbreaks and pandemics. This is supposed to be budgeted at around 64,180 crores over the next 6 years. And at NIV Pune, if this was all possible, it's because of this superb team that I have the privilege of working with. You can see we have a good section of women working. They're all huddling behind each other. I wish they had all come forward. Also men. And with that, I want to say big thank you for having invited me, and thank you for your patient listening.
Yeah. We highly appreciate and we are very proud of your contributions, Professor Priya Abraham, for validating the kits, conducting zero essays, transferring technologies to so many companies. And not only in the country, you have also tried to help other countries. We are very, very proud of you. And thank you for your excellent talk. Maybe you have several questions awaiting. We'll take it later. Over to the next speaker. Madam.
Yeah, thank you, Priya Abraham, for an excellent presentation and fantastic overview of the entire scenario of COVID. We learned a lot today. So let us now go on to the next speaker, Dr. Geeta Sharma. Geeta, I've known Geeta for a long time, and she's Director Research at Quality Control Tapadia Diagnostics at RTC Crossroads, Hyderabad. She has a very long experience. She's a consultant in biotechnology and a professor in microbiology for 32 years. And she has specialized in cloning, expression, recombinant therapeutics, and prophylactic proteins. She's founder of the microbiology department and established several microbiology departments at undergraduate and postgraduate colleges affiliated to Usmania University, and she has 50 years of research experience from 70 to 2020. So her glorious career includes plenty of research in allied areas of biotechnology at premier institutes in the United States. Like she's the PDF in Rochester School of Medicine, New York, and Memorial Sloan Kettering Cancer Research Center, visiting faculty at my Department of Virology, Mount Sinai School of Medicine, New York, visiting faculty, New Jersey School of Medicine and Dentistry, visiting faculty at Medicine University of Missouri, and a member of International Board of Virology in USA. And with this vast experience, she was able to help the industry. Like she was the principal scientist at Shanta Biotech, and she established the first rDNA biotech company in India, and she was the vice president of the biotech company Zydus Research Center, Ahmedabad, and she's director in CSO Magene Life Sciences Private Limited, the first company in India to establish a CRO to support upstream drug discovery research. And she also headed biotech Claris Life Sciences and also established the first commercial sequencing CRO lab in India. And her vast experience, she can write patents, scale up processes, and her achievements are far too many to mention here. A few of them are involved in genesis of Shanvac, the first RNA vaccine for protecting against hepatitis B virus in the country, and developed subunit vaccine for typhoid transfer, the technology to Zydus Cadila for manufacturing, and she has 50 research papers published in national and international journals. With this short introduction, I request you to kindly present your work to us. Over to you, Geeta Sharma. Unmute yourself, Geeta. Kindly unmute yourself, Geeta. We can't hear you. Geeta, please unmute yourself. We still can't hear you.
Can you share your screen, Dr. Geeta? You are the co-host, so you should be able to.
Geeta will be talking on vaccine development, past, present, and the future. Are you ready with your presentation, Geeta?
You can start your sharing your presentation meanwhile. Can you do that?
You didn't unmute yourself, Geeta, on the...
I think she's having some locking.
On bottom of the screen, there is a mic-like thing. In that, you have to unmute.
Looks like she's not able to unmute herself. Can you help her? I hope she's not muted at your end.
No, she is the meeting host. She's already made the co-host.
Because she's not able to unmute herself.
At the bottom of your screen, Geeta, you will see a mic.
And I'm sure she knows. She's a past master at these presentations.
Hare.
हेलो गीता जी, गीता शर्मा, आप अपना माइक ऑन करिए प्लीज. अनम्यूट योर माइक, गीता. We are not able to hear you.
వస్తలేదు తక్కువ తక్కువ అయిపోతుంది.
She's listening, but
ఇవేంటి తీసేవా?
unmute your mic, Geeta.
రెండు తీసేయజరా కింది పడి కష్టమైన.
She's having some problem unmuting herself.
I think the
something is
host
gone wrong.
can do it. Host can do it.
She's already unmuted, ma'am.
No,
not yet. She's already the co-host.
Geeta.
ఇట్లా అవుతుందంటే ఇది తక్కువ ఎందుకు అవుతుంది? తక్కువ ఎందుకు అవుతుంది? నాకు ఎక్కువ కావాలి కదా. 8 అయిపోతుంది. 8 అవుతుంది, ఇంకా తక్కువ అవుతుంది ఎందుకు? చార్జ్ అయితే లేదు.
Bottom of your screen, you will see on the left-hand side a mic, icon of the mic, and then you can press that and it will unmute yourself. At the bottom of your screen, Geeta.
No, I think she's having some other problem because she's.
The
Is it the
toolbars,
only-
you check your toolbars there.
Is it the only Gmail? This is the address you gave us? Same address?
She's on phone, she's talking to somebody.
Should we take the second one otherwise? The chairperson may think of calling the second speaker. Dr. Kaiser, can we take the next speaker?
Geeta is not able to unmute herself. She just called, and she's just not able to proceed. So perhaps we should go to the presentation and then come back to her. There's some
I agree. Yes, ma'am. Dr. Kaiser Jameel, can you?
Yeah, sure.
We go to the next speaker.
And can you make the introduction a bit shorter? So going very much behind time.
Madam, similar problem I too had.
I can hear you, Jyoti.
Yeah. But similar problem I too faced, ma'am, what Geeta ma'am is facing now. Somebody should unmute from the organizer's end.
They have given us the IDs, so we can do it ourselves.
Yeah.
No, you are already the co-host, madam. So I will.
Dr. Annie, can we have you as your next speaker?
Yeah. Can I invite Annie?
Yeah, sure.
Yeah, sure. Please introduce Dr. Annie.
Yes. We have the next speaker, Dr. Qurratullah Hassan. She's very much known as Dr. Annie. She's a best friend of mine. And she's a senior consultant and head department of genetics and molecular medicine, Kamineni Hospitals. And she's the principal of DNP courses at the same hospital. And she has guided 21 PhD students. She has more than 135 national and international publications. She has started the first full-time postgraduate certificate course in medical and genetic counseling in 2007. And Dr. Hassan is the founder member and president of the Board of Genetic Counseling India from 2014 till date. May I now welcome Dr. Annie.
Thank you, Jyoti, for the lovely words, and thank you to the organizers, especially Geeta, for inviting me to give my presentation.
Pleasure, Annie.
So I'm trying to share my presentation. I'm not able to click share. Yeah.
हाँ दीदी.
Can you see mine?
गैस बंद किया ना आपने?
Hello.
Madam, you have to click on the presentation. Open the presentation and click on that.
Yeah, I can see the presentation, but you all can't see it?
No, we can't see.
Okay. So,
no, we couldn't see.
You can't see it? Okay.
It says here you are presenting to everyone. Yeah.
Just try again, and if you have an ignore button, please click the ignore one.
No, when I'm clicking, it just says cancel. I'm not able to share it. 1 minute.
Open your presentation.
It's open. It's open.
Okay.
My presentation. What should I say? 1 minute.
Ma'am, please don't click on share screen. Click on share tab. This is my suggestion.
Dr. Geeta, maybe you can log in again and try. Meanwhile, Doctor.
I don't know if Dr. Sudhamurthy is ready.
Sudha, did you try? Can you come on screen, Sudha?
Yes,
I'm ready. I'm ready.
Just try if you can. We can hear you. Can you present your screen?
Shall I share my screen?
Yeah, you are already. All three of you are the hosts.
Okay, I'll just try.
Yeah, it's Annie's has come now.
Annie's has come. Yeah.
Oh, sorry.
I'll stop.
Yeah, Annie can please go ahead.
Yeah, Dr. Annie's is coming.
She has to unmute, ma'am.
Yeah, that's true.
अभी अनम्यूट हो गया?
हाँ, हो गया गीता मैम का भी आ गया.
Can you hear me?
Yeah, we can hear you.
And you can see my slides?
Yes, Annie.
Hello.
Yeah,
Can everybody hear me? Am I unmuted?
Yeah.
Yes.
Okay, thanks. I'll not talk, but let it remain.
Can you all hear me?
Yes, Dr. Annie.
But I am not able to see my slides.
You want to see a slide?
Then it will be pressure.
Chalo, Annie. Talk now.
This is whole thing is here. Okay, I'm not able. If I do the full screen, then I'm not able to see my slides. So I'll do it this way only. Okay, is that okay?
Put it on slideshow, no, Annie?
It is not coming. If I do that, then I'm not able to see my slides.
Okay.
Do it again. It's not coming back yet.
Your slide has appeared, but it is in the
Usually we
box.
have seen your slides when both yours and Geeta's assignment came up simultaneously.
Why the two in? एक मिनट नहीं. Okay. So what's happening here?
Now, are my slides visible?
Not at all.
No. Okay.
चलो जी, give me a second. Just बोलो. I'm taking a look at this. 1 second, please. I'll just select a tab.
Geeta ma'am, are you ready?
Wait.
Geeta Sharma?
Oh God, she's on phone.
No, I think Annie's slide should come because earlier there were 2 windows.
Not a share. Not a window.
If both of you are taking time, we can start with Dr. Sudhamurthy. Can we?
Yeah, Annie's slides have come.
Come, yeah, okay.
Where is this?
You know, when I'm able to see the slide, you all are not able to see it. I don't know what it means.
You have to make a reverse thing there.
Have you?
Why don't you just talk? Can you see the thing? Yeah, just keep talking.
Can you see the slides?
No, Annie.
Not yet open. We can see your name there. Your presentation slot is there.
Okay.
Is it possible for you to send us the slides, Dr. Geeta, Annie, and also Sudha, just as a backup?
Share a tab. Select a tab.
Okay.
What is this?
Sudha, are you ready with your presentation? Oh yeah, now Annie is coming.
Yeah, now I can see Annie's.
Yeah.
Make it full screen, please. And please make it full screen.
Annie, the slides are seen now?
Yeah, so you can hear me?
Yes.
Yes.
Yeah, I just cannot do full screen because every time I'm doing full screen, it's just going. I think that was
we can see the slides. We'll manage this.
Yeah,
way.
we'll just do it with it. Okay.
You can make the slides sides bars smaller if that helps. Otherwise, leave it. We'll just go that way.
Yeah, we'll just go ahead. Just let's forget it.
Yes.
Okay. So the title given to me is Cytogenetics to Genomics, Advances in Technology. What I thought, because the not
Okay, I can't
moving.
hear.
It can't move. It's not moving. Up to wait. Go to click on this.
Just click on that.
I've sorted out if you want.
Yeah, hey, it's not moving.
It's not moving.
Okay, let the next person
Big
present.
bar.
No, it's not happening.
Sidebar.
No.
Yes,
Not.
Next slide. The sidebar. Click on the slide itself, madam.
I'm clicking on the slide. No. Only the first.
Geeta, I think I'm ready. Shall I go ahead then?
Yes, please go.
Yes,
Okay.
ma'am. Please.
Okay. Now I'm trying to share my slide.
Yeah.
We have to remove cytogenetic slide.
Yeah, that I'm not able to do. Now, how do I do that?
Maybe Annie has to close it.
Annie, can you pause yours so that I can put mine?
She did it. Now you go ahead.
I have done it.
Okay. Okay, it's here. How do I maximize share? No, But now I'm not seen, right?
No, we are able to see you, ma'am.
Now, how do I get both of us on the screen? 1 minute,
Put it on slideshow, ma'am.
Okay.
Click the first slide, madam. It will automatically come. Double click.
Yeah.
Yes. Yes.
Okay, but I'm not able to see myself on your screen. Is
That's okay. We can see you.
Okay. All right. Good. Okay. Thank you very much, Geeta from University of Hyderabad for inviting me. And Kaiser, thank you too for giving me such an elaborate presentation. Without doing any further delay, let's get on to the talk itself. Now, if you look at the origin of, my topic was vaccine past, present, and future. So speaking of the past, Edward Jenner was the first person who introduced vaccine, and that was smallpox vaccine in 1796. And like one of the speakers was saying, anything that's done for the first time is ridiculed. In fact, Edward Jenner was stoned, thrown, whatever else. And finally, the vaccine became accepted. Then from there, in 1982, the first recombinant product ever to be licensed for manufacturing was Humulin or insulin, recombinant insulin that was marketed by Genentech, the first recombinant DNA technology to be launched in the market. But even before this, what happened was Bloomberg in 1981 had the approval for the first hepatitis vaccine, which was serum derived. Subsequent to this, 1 year later, it was in 82 that a recombinant DNA technology product was accepted. Soon after this, in 1986, Recombivax was the first one. This was in 82 and this was in 86 that it was launched. Then the Recombivax was withdrawn for certain reasons, and finally that came into market was Energix B in 1989. And we in India were not far from the Western country, though we were at that time a developing country. In just about a decade later, we in India launched Shanvac, the first hepatitis B vaccine in country, as well as the first recombinant DNA vaccine or DNA product in our country. So this was a moment to cherish, because though we were labeled as a developing country, we came caught on in par with the developed countries. And just in about less than a decade, we also launched a novel hepatitis B recombinant vaccine, and it definitely was a moment to cherish. And from then, it was a long travel with its struggles. Today, I shall walk all of us through the landmarks that led us to the battles against those enemies, whether it's bacteria or virus. Virus, for that matter, is only 20 nanometers to 400 nanometers. And that small little particle is able to shake us up, which is of so small in dimension. So if we are speaking of what are the different vaccine strategies with the virus, mostly I'll give virus as an example, but what holds good for the virus also holds good for the bacterial vaccines. So when we speak of the different strategies for vaccine, one alternate could be an inactivated virus, or we can have an attenuated virus as a vaccine, subunit, virus-like particle, DNA vaccine, RNA vaccine, etc. So there are 7 types of vaccines that we can broadly classify. Live attenuated, which is what is even our Bharat Biotech vaccine. And the major advantage of this versus the DNA vaccine will be my concluding slide. Apart from after live attenuated vaccine comes the killed or inactivated vaccine, then a subunit, toxoid, conjugate, recombinant protein, nucleic acid vaccines, etc. So in a live attenuated vaccine, what happens is the disease causing agent is weakened in the laboratory, wherein it slowly loses or its capacity to multiply and go into the infective cycle becomes very poor. Nevertheless, it has a full immune response. It is capable of inducing the T cell, helper T cell, and the complete immune system. However, there is a small risk along with all the advantages, in that it might revert back, and in very rare cases, the person might come up with the disease. In killed vaccines, the organism is killed by chemical, heat, radiation, antibiotics, etc. And in this, the chances that it will ever revert is not at all there. It's very safe. However, it cannot induce killer T cells, so it may not work for certain pathogens. So the examples of these are Haemophilus influenza, cholera, polio, Hep, rabies vaccine, etc. Toxoids are again most of the bacterial diseases that we get essentially are from the toxoids, or a substance that the bacteria secretes into the system. And this toxin that it secretes is the one which is harmful product. And therefore, if we could use this toxin itself to create immunity, then that would be a fair enough opportunity. So we have methods where we extract the toxoid, inactivate, purify it, and administer that as the vaccine. Subunit vaccine, again, instead of the whole vaccine, a part of instead of the whole organism, we take only a part of the organism which is capable of inducing an immune response. For example, in typhoid vaccine, it is the polysaccharide that causes the immune response. So in a typhoid vaccine that I created for Zydus cadila was where I took the polysaccharide unit from Salmonella typhimurium, and we purify it and administer that as a vaccine. So for all these subunit vaccines that we do, like in this case a polysaccharide, in some other case it could be another part of the organism. So this is tagged on to a carrier molecule which is fairly immunogenic, and such vaccines are called conjugate vaccines, and that also is a possibility. Or one could just extract the membrane of the organism which has the antigenic recognition sites present on the outer membrane vesicles as blebs, and these are purified and modified so that the toxic portions are removed, and it is administered as a vaccine. For example, in meningitis B vaccine is one such. Then those were the first generation vaccines wherein the whole organisms itself were used, or part of the whole organism was used. Then in the second vaccines is the recombinant vaccine, wherein we genetically engineer the organism and the antigenic. For example, let me explain with hepatitis B, which is what I have done for Shanta Biotech. Now, the earlier hepatitis B vaccine, which was Embrax, was done with the hepatitis B pre-S as well as the S antigen, and it was grown in yeast cells, that is Saccharomyces cerevisiae. However, in Shanvac, what I did was knocked off the pre-S region, took only the S region, and instead of using Saccharomyces cerevisiae yeast, we used Pichia pastoris as a yeast, and then we grew the okay, first you make a construct. The construct the plasmid over here will have a 1 minute.
Your slides are not moving.
It's moving now?
Still at the live attenuated vaccine.
Okay. Okay, coming back to the hepatitis B protein vaccine. For example, from the viral gene, is the slide visible? Hello? Hello?
You are still at the live attenuated vaccine.
Oh my.
Your slides haven't moved.
Oh, what do I do then? Wait a minute.
Yeah, now see, when you're moving that sidebar, it's moving.
Okay, okay. I'll do it here then.
Yeah, that's better.
Okay. So we did the attenuated vaccine. Then I spoke about the killed vaccines.
Oh, yes. Now they're moving.
The toxoid.
Yeah.
In the toxoid vaccines, what one does is we take only the toxin that the organism produces. And from this, the toxin is inactivated, whatever, and then centrifuged, purified, and then administered. The examples for this is tetanus, which is done in horse serum. Clostridium is the organism that produces diphtheria toxin, then Clostridium botulinum, pertussis. All these are the toxoids that cause the disease. So the organism is grown in other animals. The toxin is purified, then it is inactivated and administered. In subunit, instead of, pardon me? In subunit vaccine, a portion of the organism only is immunogenic. It could be the polysaccharide, it could be any other portion of the organism. For example, in typhoid, which is what I did for Zydus cadila, and it's in the market by name Tyvac. What we did is isolated the polysaccharide of the Salmonella typhimurium, and this sugar is conjugated with the carrier molecule and it is administered. Okay, and here what happens is the advantages of conjugate vaccines are that it has not only the ability to elicit the immunological memory, it also reduces the asymptomatic carriage of the bacteria, resulting in herd immunity. Now, in OMV vaccines, what happens is for the antigenic or the immunogenic component in the organism is in the outermost shell of the organism. What happened? Hello? Hello?
You're audible, ma'am.
Okay.
Disturbance, you're audible, but may I request you to, I'm sorry, because we are running late, we can shorten your talk, please. Thanks.
Pardon me? Shorten. Okay. So those three were all the first generation vaccines, and the recombinant protein administration as a vaccine is from the second generation. And this is where most of the vaccines are administered even today. And this category is what hepatitis vaccine is. So over here, what was done is the viral gene, instead of taking the full pre-S and S region in Hep B vaccine of Shanvac, we've taken only the S gene, we put it into a plasmid, and this whole thing is constructed. This is an expression plasmid wherein we put this gene. I'm not going into too many details as there is a shortage of time. Okay. So this is the detail. I mean, once the gene or the immunogen is put, it's inserted, that is again transformed into the yeast, and this is again this is not moving now. Okay. So the protein which is expressed in yeast is now purified and then administered. So that is a protein vaccine. Now the third generation vaccine that we are speaking of are the DNA vaccines. In the DNA vaccines are made up of the plasmid itself, and the plasmid is what is injected into the human being as a vaccine. So the injection of DNA is done either in saline or by gene gun delivery. And the cell's host machinery now from this plasmid that is entered the human host, the host cell now synthesizes the protein of the immunogen, and that the human body recognizes and triggers an immune response. So if it's an RNA virus for which we are making a DNA vaccine, the genomic RNA is extracted. From that, the genomic RNA from the virus is extracted, and its cDNA is made, a double-stranded DNA is made.
Dr. Geeta, you have 2 minutes left for your presentation.
Oh.
Yeah. Thank you.
I don't know then what to do. Okay, the advantages of, I don't know. I just give up.
You can talk about the future, ma'am.
See, whether it's RNA vaccine is what we are using today, wherein the RNA is extracted from the virus, and that is packaged into a microparticle, and that is administered into the human body, and that now makes the antigen. So the future of the DNA vaccines are, like, the transfixion of myocytes can be amplified by pre-treatment of the muscle structure itself, muscle itself, and so that the myoblast regeneration occurs. And to understand this whole cycle properly will enable us to see that maximum amount of DNA uptake occurs. And this kind of a research would also enable us for gene therapy capabilities. In RNA vaccines, the messenger RNA is packaged into a lipid fat membrane, and this lipid body molecule, along with the packaged RNA, is injected. So, for example, this is the spike messenger RNA is isolated, and then it's put into a lipid cell, and that is injected. And once it goes into the body, the messenger RNA now makes the spike protein, and the spike protein is recognized by the immune system, and antibodies are produced. The other alternative, or the third generation, are the viral vectored vaccines, wherein nucleic acids are, non-infectious viruses or animal viruses such as chimpanzee or something which do not infect human beings are used as vectors, and the viral RNA or DNA for that matter are packaged into this viral vectors, and this the absorption becomes easier, and that is administered as the vaccine. Like there is a trial going on of chimpanzee adenovirus, wherein COVID-19 RNA is being used, and on the one hand, this construct is being made where the COVID RNA is present here, is taken, the spike RNA is taken and put into the chimpanzee adenovirus, and this adenovirus is now injected into the human being, and they make the antibodies. I think this will be newspaper information, I don't know, wherein which company is making which vaccine for COVID, etc. So if anybody has questions, I'll answer in the question time. Thank you very much.
Thank you, Geeta, for the overview of vaccine development and all. Sorry, we had technical problems and you had
because over here, the slide is not coming in. On my computer I have no eye contact. I have no, I don't know.
We are really missing your excellent presentation, madam. We'll
no.
have it once again.
You have so much content with you that, I don't think we can absorb so much content.
Geeta, that's very polite of you.
No, I mean it, because you've spanned the entire virus spectrum.
I agree. Yeah.
All right.
Dr. Annie will be ready now, I think.
Geeta?
Yes.
Will you be sharing the presentation?
Why will I share the presentation?
I sent it by email to you, or do you want me to do it?
That Geeta. Okay, I'm sorry.
Yeah.
Open the presentation.
I think we have a lot of Geetas today. So very nice.
So Geeta, will you be presenting for me?
Yes, we are doing that. Yeah.
Okay. So I'll be switching gears and not talking about SARS-CoV-2 or vaccination. I'm going to talk about the development of techniques which started for genetic testing, from cytogenetics to the molecular. So starting from the very beginning, every cell has chromosomes, and human beings have 46 chromosomes. And funnily, DNA was discovered much before the chromosome number. So chromosome number as 46 was established in 1956, whereas, yeah, should I just say when to change the slide? Yeah. So it was in 1956, we came to know that there were 46 chromosomes in the human being. And it was in 1953 that we knew that DNA made the chromosomes. And segments of DNA are the genes. So study of the chromosome, next slide please. The chromosome number and the structure was cytogenetics. So studying cytogenetics helped us to identify Down syndrome, Patau's, all the major syndromes. And it also helped us to understand a lot about infertility, where there were translocations, which is on the right side slide. You can see that chromosome 1 and 7, there's a translocation. So when they are passed on to the next generation, there's either a duplication or a deletion. Now, next slide. Cytogenetics helps us understand pregnancy loss, infertility, developmental delay, genital ambiguity, and a lot of hematological cancers. But cytogenetics and molecular biology together developed fluorescent in situ hybridization, the next technology. Next slide, please. Where different regions of the chromosomes could be identified, and this could be identified without culturing and getting mitotic cells. We could do it in interface cells. And that is why amniocentesis and all could be done very quickly, where you could just put the cells on the slide, put the regions of interest, like most commonly chromosome 13, 18, and 21, which resulted in Edwards, Patau's, and Down syndrome. And as you can see on top, you see 2 signals of each, but in the bottom slide, you can see 3 signals of the red saying it's abnormal and it's responsible for Down syndrome. Similarly, fish, next slide, played a very important role in cancer, and you could take tissue blocks or take bone marrow cells or hematological cells, and without looking at the chromosomes exactly, we could say that there has been a chromosomal translocation or amplification. So on the right, you see breast cancer tissue, paraffin blocks, and you can see the red signal increased, and the red here is HER2. Below, you can see chromosome 9 and 22, a translocation between chromosome 9 and 22 results in a BCR-able fusion protein, which can be seen as a red, a green, and a yellow. A yellow signal indicates fusion. So FISH was a technique which could tell us specific regions. But when we further advance to microarray in the next slide, you will see that small regions of the chromosome could be studied as microdeletions or microduplications responsible for various genetic disorders, like D. George giving chromosome 22 deletions, Wolfram's chromosome 4, and so on. So small regions which were deleted or duplicated, which could not be seen in a simple karyotype, could be seen by chromosomal microarray. And chromosomal microarray has now become the first line of testing, both in utero and of individuals which have multiple anomalies, but we do not define them as a particular chromosomal type. When we go further to molecular techniques, next slide, please. DNA isolation and storage methods, as they advanced, we came to do a lot of molecular testing. And the development of PCR, polymerase chain reaction, is pivotal to all these molecular methods, be it single gene, multiple gene, or single nucleotide changes. Next slide, please. In this, we have shown that one single gene, MTHFR, which is responsible for the folate pathway, if that has a variant, a single nucleotide variant, you can do a PCR and you can do a RFLP or you can do a Sanger sequencing. So RFLP gives you two bands if one allele is normal, one is mutated. And similarly, you can see the same thing as two heterozygous peak in the middle chromatogram. So, but the technique is based on PCR. So first you take the DNA or any tissue from which you can get the DNA and are able to get the RFLP or the Sanger sequencing. When we go down ahead, next slide, please. Can you go to the next slide? Okay. I don't know why. Anyway, if you can hear me, I'll continue and tell that now with the advent of NGS, what has happened is that instead of looking at a single gene, we are able to look at panel of genes simultaneously. We are able to do the whole exome or the whole genome. So the advances in technology has helped you to come from looking at chromosomes, which are 46, to the 22,000 genes, which we are able to assess in one test. Now the question is, which test is required for what purpose? And there comes the point of choosing the right test. So when you are looking, if you are trying to see pharmacogenomics or individualized medicine for a person who's taking multiple drugs, the person is taking a drug for hypertension, taking a drug for sleeping, insomnia, or taking a drug, a proton pump inhibitor, because of the acidity these drugs are causing. So you need to look at several genes and several variants, and that is where we come called array-based testing. And this array-based testing is really important for people as they age and they are dependent on multiple drugs. Next slide. The best of Sanger sequencing cannot tell you about deletion duplications which are large, like for Duchenne muscular dystrophy or for individuals who have BRCA mutations which are large deletions. And then what we need is MLPA. MLPA helps us look for large exonic deletions. And for this, remember, NGS doesn't help. Although we consider NGS to be the most advanced, it does not help us. Next slide. So NGS-based tests, like I told you, without the slides, were this. So now I'll go to the next slide. The cell-free DNA testing, where this I think Sudha will be dealing with, so I will not talk much about it because this is playing a very important role in cancer biology and cancer advanced testing, where it is a non-invasive method of testing. And we have heard about the chromosomal and the biochemical testing for cancer earlier, alpha fetoproteins and all. And now we are directly looking at the cell-free DNA of the tumor in an individual's sample. Next slide is the methylation. Now, not only the tests of the DNA, but how the DNA is methylated is becoming more and more important for diagnosis. And this plays a very important role in the next slide in imprinting disorders. For years, we have believed that both the mother and the father contribute equally to an embryo. But we realize now that there are 200 genes which are differentially expressed from the parents. If they are expressed from the mother, they are silenced from the father. And if they are expressed from the father, they are silenced from the mother. And this is mostly known as if alterations in this occur, then we get imprinting disorders. And imprinting disorders cannot be detected by NGS or FISH or Sanger or karyotyping, but we require a methylation specific PCR. So last slide is about the summary of the test timeline. So in the 1960s and 70s, cytogenetics was the only test available. Subsequent to that, in the 1980s and 90s, PCR came into play. So molecular tests became very important. And then after 2003 to 2014, when the Human Genome Project came in and NGS technology increased, we could do the panel tests and exome tests. And now we have the whole repertoire along with MLPA and epigenetic testing for us to understand several genetic tests. But now the difficulty is in choosing the right test. So which is the most appropriate test for which genetic disorder? In the next slide, I end and I thank the genetic counselors who are there to help clinicians, patients, and families to choose the right test. So I will not take too much time, and I would end now with thanking you all for giving me this opportunity.
Oh, very clear and lucid talk, Annie.
Thank you so much.
In fact, we used to think that these are research tools, but by the time we started hearing about these tools, they actually became, they came into the clinics.
Yes. So it happened quickly. And since 2015, we are using it in the healthcare sector. So when people say genetic tests will be used in the future, I get very upset. No, they are already there. And they are being used for both personalized medicine, oncology, intellectual disability, and infertility.
Okay, wonderful, wonderful. So in two of the ocular tumors, in retinoblastoma, our detection rate was initially 50 when the Canadian group was almost reaching 80 to 90. But with addition of more and more techniques, now it's reaching 80. And as you rightly said, one technique is not enough, at least for retinoblastoma. Yes. There are multiple, complementary tests that you need to do to get the entire screening and rule out all the mutations, because it's a very large gene.
MLPA also is used for retinoblastoma, breast cancer, and Duchenne. I think that works out and gives you the whole picture.
Yeah. So in uveal melanoma, it's now, we don't have uveal melanomas here, but in in Caucasians, from a biopsy, it's a routine thing to do the genetic testing and classify them based on the microarray and the monosomy 3 into class 1 and class 2 prognostic. Yeah, is over to you.
Hello, Geeta. Hi. Thank you.
Could there be a tiny question?
Sure. If it's for me, sure.
Yeah, for you, yes. What are the new panels for prenatal genetic tests which are coming into the laboratory space and clinical space after the entire information has opened up through the exome panels and NGS?
Yeah, we still, now we recommend for amniocentesis, we do a microarray in which we look at the 3 commonest aneuploidies, which is done all the time, 13, 18, 21, because they still remain the most common, followed by 13 deletion duplications of microdeletions for D. George, Williams, Cry-du-Chat, Wolfram, and so on, which are very common. We also do PWS and Angelman, 15Q. So it's a microarray panel which we offer for prenatal. But if it is, if we know of a molecular thing running in the family, like Geeta said about retinoblastoma, then we do the targeted DNA analysis for the mutation which we have identified in another SIB or in one of the parents.
Okay. My question was actually more related to the frequency of increases in congenital diseases and whether panels are now coming out for those
thalassemia, cystic fibrosis, we include now, but then we do it with molecular first in prenatally in the mother and then prenatally.
Okay, thank you very much.
Jyoti, you have to unmute.
You're done now, Annie? Thank you
I'm
so
finished.
much.
Thank
I
you.
know there was a power cut, so couldn't follow the last part. Anyway, I'm happy to introduce the next speaker, Dr. Sudha Murthy. Sudha, I'm so happy to introduce you. You were formerly my colleague and friend when I was at the Basava Tarakam Indo-American Cancer Hospital and Research Center, 2002 to 2006. So we were together for some time. So I'm happy to see your new profile as Director of Data Cancer Genetics, and you're heading there, and your profile shows that you're oncopathologist and healthcare management professional and avid speaker and passionate teacher. You have 82 publications in peer-reviewed and national and international journals, and you have received Best Teacher Award, DNB Pathology, and also you have oration and honors like you have in molecular pathology, diagnostics, and immuno-oncology. And also gynec pathology and soft tissue sarcomas. You have great experience in these areas. And you have done your ISB at Indian School of Business and Advanced Management Program in Healthcare. And you were at Nizam's Institute of Medical Sciences, where you did MD Pathology, DNB, MNAMS, and also
that's it, madam. Thank you.
I think that's it. Okay. And you have membership in many academies in pathologies and Indian School of Business and all that. And then
thank you, ma'am.
I'm just handing over the mic to you, Dr. Sudha Murthy.
Thank you so much, ma'am. Thanks for the kind introduction. At the outset, I would like to thank Dr. Geeta. I hope my screen is visible.
Yeah. I'm moving your slides.
You can see my
liquid
slides?
biopsy.
Okay.
You can see
I'm moving
your slides.
Yeah. Thank you so much, Dr. Geeta, IWSA, and University of Hyderabad, Jyoti ma'am. Thanks to everyone. Dr. Annie Hassan has set the stage, so I can quickly go through my slides. So
I'm happy that my junior from NIMS has reached this stage. And
oh my God, madam, you are an inspiration. You are an inspiration to all the alumni of NIMS.
Thank you.
So we know that tissue acquisition and analysis remains the gold standard for diagnosis and now, of course, for molecular profiling. However,
can you speak louder?
Yeah, sure, ma'am. Am I audible now?
Yes, ma'am.
Am I audible?
Yes, you're audible.
Okay. Yeah. So, but now we know that novel methods by utilizing liquid biopsies are now available, and these are hot topics for research and also have reached the bench side. Now, especially during the pandemic, we have seen that these liquid biopsies have gained substantial traction because patients were not able to reach their doctors,
And that is where they found this very valuable. The clinicians found it very valuable because of the reduced turnaround times and the hassles of an invasive procedure. But let me make a statement. I am a histopathologist, so it is a complementary tool, and a pathologist would understand how it can benefit in both ways. How tissue sequencing still remains very, very relevant, and liquid biopsies also contribute significantly. So now the clinician is confronted with several situations, as madam had, my predecessor, Dr. Annie, had identified, that the clinician needs to know which ones of these tests have to be ordered. So whether it is liquid biopsy, whether they need to go for an exome sequencing, or they need to go for a smaller targeted panel. So these are the dilemmas that are coming, and therefore there's a need for a lot of awareness and education even amongst us clinicians. So liquid biopsy, as we all know, it has applications in organ transplants, oncology, and IPT. But today we'll restrict our discussion to oncology because this itself is a big ocean. Liquid biopsy is nothing but non-invasive sampling. That is, it could be any liquid for that matter, blood, any of the fluids, peritoneal, pleural, pericardial fluids, sputum, urine. But since liquid biopsy needs a lot of validation, it becomes difficult for labs to use all of these samples because each sample needs extensive validation, which costs quite a bit. And we know that liquid biopsy is a consequence of two advances. One is human genome sequencing, and there has been tremendous innovation in the diagnostic space with focus on increasingly sensitive, non-invasive assays. Now, when we look at the liquid biopsy, it is not just one element. It's not just cell-free DNA or circulating tumor DNA, but it also encompasses circulating tumor cells, exosomes, microRNAs, methylation studies, extracellular vesicles, and the list goes on. Now we have tumor-educated platelets. So all of these elements are being studied and trying to find out which one of these would be useful at the bedside. And that is where we are trying to make our patient's life easier and accessible. So these are the different elements of liquid biopsy. We see that CTC and ctDNA already have reached the bedside. People are ordering clinically, clinicians want these tests. Circulating microRNAs, as most of you would be knowing, that Datar also has its international labs in London and Boston. So there we have traction for circulating miRNAs, microRNAs, which has prognostic relevance. At this point in time, doesn't have diagnostic relevance, and extracellular vesicles. So liquid biopsy, as per ASCO-CAP definition, is a broad category for a minimally invasive test on a sample of blood to look for circulating cancer cells or fragments of tumor-derived DNA or RNA. So this is broadly defining only the ctDNA and the CTCs. This is a pictorial which was published way back in 2014. This is nothing new. Actually, CTCs have been identified nearly 40-50 years ago. But we did not have sensitive techniques or the awareness to make use of these very important components of the liquid for the benefit of patients. So for the circulating tumor DNA, we know that it is a component. Cell-free DNA is the entire DNA that comes from not only tumor cells, non-tumor cells like WBCs, macrophages, etc. Circulating tumor DNA is a component of cell-free DNA, which is a very small constituent, and therefore these assays are not said to be as sensitive as the tissue DNA. So nevertheless, especially in the early stages, but with the improved techniques, improved methodologies, excellent awareness and research programs going on across the world, we have seen that this has caught up traction even in the oncology space. So as we all know, this cell-free DNA, it can be sampled in an idiot tube, but then you'll have to really immediately centrifuges. Therefore, now we have this Streck tubes, which have preservatives, and they can keep the cells, the DNA viable for a longer period of time. Exosomes, I will not dwell much upon this. It hasn't reached to the bedside to that extent. So how does the ctDNA? So we all know that tumor cells, they undergo apoptosis, necrosis, and secretion. And these elements come into the blood, wherein we can evaluate, we can isolate them, and then we can quantify them and also look at the gene expression or mutations on these. So the different components will not dwell much upon the research elements. This is the poster child for liquid biopsy. So lung cancer, as we know, all the pathologists and all of my colleagues here in the panel would know that lung cancer is a very difficult biopsy to obtain. You get very small tissue and the pathologist is asked to do a host of tests on the small tissue that is available. You have to give the morphologic diagnosis. You have to do an IHC to ascertain which type of carcinoma it is. You also have to do with the immuno-oncology, advent of immuno-oncology, you have to do a PDL1. And then of course, all of these genes, the therapeutic targets, which are essential today for treatment. So how does, tissue sequencing, we know it only gives a single snapshot of a particular tumor region. So when you're sampling something, it is only looking at that particular region. But we know that there's a lot of heterogeneity, whether it is spatial or temporal. When we are talking of spatial heterogeneity, it is heterogeneity, difference in the expression or the mutations between the different sites of the same tumor or between the primary and metastatic tumor. When we are talking of temporal heterogeneity, what would have been initially at the time of diagnosis would change after treatment. So liquid biopsy is a tool which will take care of both. So you have a baseline, and even if the dynamics have changed over a period of time, liquid biopsy you can repeat easily and get a what is the change that has happened so that the oncologist can address that particular change. So this is a very landmark article from Nature which shows the different subclones which cause the heterogeneity. So this is the reason why when we sample a small amount of tissue, probably we are not looking at the whole picture, and that is where liquid biopsy comes very handy. So these are different papers which have demonstrated intratumoral and intermetastatic clonal heterogeneity. These are the differences. All of us know it's an invasive procedure. The liquid biopsy is non-invasive. It can be easily repeated. Still, gold standard is tissue biopsy sequencing because I told you the sensitivity is lower for the liquid biopsies. But now the things are changing. The ctDNA, also the liquid biopsy picks up subclonal mutations, which might not be picked up on your tissue sequencing. These are the different technologies. Madam has already alluded to all of these, RT-PCRs, droplet digital, and next-gen sequencing. But now that we know the larger panels are what are required, you cannot address them with RT-PCR or droplet digital PCR. So we know that ctDNA levels are low in most early stage tumors or post-treatment patients. And therefore, for liquid biopsy, you have to do ultra-deep sequencing. And that is where cost is concerned. We see these challenges on a regulatory perspective. Everyone says, I have a liquid biopsy, but we are not achieving the optimal depth. So there's a chance of false negatives. So quality is very important. And chip is something which is a caveat in liquid biopsy. It is nothing but clonal hematopoiesis of indeterminate potential, which is age-related expansion of normal hematopoietic cells which harbor somatic mutations. So this pictorial shows you the child does not have any mutations, but as age passes, the individual has developed passenger mutations, which might be mistaken as mutations on liquid biopsy. So this we have to be wary, and therefore the filters and bioinformatics pipelines are very important. Excuse me. So false negative result may arise at several steps: hemolysis during collection, low depth of sequencing and coverage, inappropriate filters applied to bioinformatics data, and erroneous interpretation of data. Therefore, in all of these assays, quality is the baseline parameter. Every step you have to have very stringent quality. So liquid biopsies are looking at screening, diagnosis, prognosis, therapeutics, recurrence monitoring. They are reaching everywhere. And we are glad to inform that we at Datar also are looking at each aspect of this. And we are trying to see if our Indian population would have the benefit of this. Now, switching gears, I move to circulating tumor cells. These are malignant cells which are shed by the tumors into circulation. So gone is the belief that we used to, when we were in medical school, we used to believe that once it goes into circulation, it is a stage 4 disease. Now it is conclusively proven that circulating tumor cells are shed into the circulation even when the tumor is very early and not detectable. So that it's equivalent to microbiopsy and can be highlighted by EpCAM positivity, pancytokeratin, CD45 negativity. So this was defined by a technique called cell search, wherein they had isolated the circulating tumor cells and then tried to phenotype these cells with these markers. But then cell search was found to be less sensitive, and researchers started still working on better sensitive assays. In addition to CTCs, the clusters are also important, which carry the phenotype and the genotype of these tumors. So these have been also utilized to improve your sensitivity. So I'll just give you a small example of a test which has received US FDA breakthrough designation. We are proud that India represents the first country to have got US FDA breakthrough designation status in the CTC area for symptomatic triaging and also for screening. And proudly, Datar is the lab which has done it. So this is on the patients who have elevated PSA, suspicious findings on digital rectal examination, and ultrasound shows enlarged prostate. Now, in a conventional approach, what happens is we have patients who undergo biopsies. Some of them would have malignancy, some of them do not have. And 25% only are malignant, especially in countries like UK, where there is a good screening program. You find that only 25%. So unnecessarily, we have biopsied 75%. So if you have a tool, blood-based tool, which can triage and help the urologist ascertain which ones have to go for biopsy, which should not, it makes it very easy. And that's where CTCs come into the picture. So we know that millions of prostate biopsies are performed every year. Now, what our solution or the CTC-based solution does is it segregates those people, and those who do come positive for the CTCs specific to prostatic adenocarcinoma, we are very clear that we are looking at only 95% of cancers. We are not looking at the sarcomas, lymphomas, etc. We are looking at prostatic adenocarcinomas. So if it comes positive, then you have to go for the standard of care methodologies like a trust-guided biopsy. Obviously, CTC cannot replace a biopsy because it doesn't give the Gleason's grade grouping, the perineural invasion, and other prognostic parameters which are needed for management. So this is only helping triage and reduce the pain or the other complications of an invasive procedure. So this is a useful tool for selection of patients. Now, just to give you an overview of how it goes, you take symptomatic individuals, peripheral blood is taken, negative enrichment is done with a media, and then the circulating tumor cells are isolated, and these are dissociated. The clusters are isolated, they're dissociated into single CTCs, and then they undergo ICC profiling, the immunocytochemistry profiling, which are tagged with fluorescence tagged antibodies. So once you have positive circulating tumor cell, they are stained with specific organ-specific markers, like for example, in prostate, PSMA, AMACA. And then we say that this is negative or this is positive. And then the urologist is advised to go further biopsy is needed. This is how the images. May I request someone who's unmuted to please mute? So these are the pictures, the fluorescence images, which show the staining, and this is captured with high content imaging platforms. So it is not just subjective. You get an objective analysis, an image analysis of all of these cells based on the different parameters, and then based on the cutoffs that are established during validation, you say that this is positive or negative. So please mind it that for screening, the cutoffs are different. For the symptomatic triaging, so it's very important to understand what is the status of the patient. So this is available directly to the customer for the screening, but for patients who are symptomatic, it is through a doctor's prescription, because this step is very important to identify which solution the patient needs. So this is the breakthrough designation which has come recently for the early stage prostate cancer detection blood test. Way back in 3 months ago, we also got for the screening, the early screening of early stage breast cancers. Even DCIs was there in the cohort, and US FDA breakthrough designation was given for what is marketed as Trinetra in USA and EasyCheck in India. So these are just the figures to show that CTCs are more sensitive, especially in screening, than ctDNA. So I'll be happy to share. So these are the publications with reference to that. Our group had shown that hallmark circulating tumor cells and their clusters, they signify 230 times higher 1-year cancer risk. Some of these were groundbreaking. And here we had also shown that the tissue immunohistochemistry corresponds with CTC immunocytochemistry. And This was the recent ASCO, wherein we had presented the concordance of immunocytochemistry profiling of CTCs with immunohistochemistry on tissues for different organs. So beyond CTC enumeration, what else do you get from it? We also do the other theranostic tests like ERPR, HER2 on the CTCs. You can do androgen receptor variant 7 detection to predict androgen resistance in prostatic cancers. And this is very handy for oncologists and the urologists. You can do PD-L1 expression, single cell studies, genomics, you can do on the single cells. And of course, if you add the sarcoma markers, you can identify the non-epithelial malignancies. Lymphoma, it's still a hurdle. We are not able to do that. So in addition, CTCs also can help in the prognosis of various tumors. In the interest of time, I'll not go through this, but what has been established already part of guidelines is breast cancer, prostate cancer, and colorectal cancer. Pancreatic cancer has a dismal prognosis, and this is again one area where people are looking at opportunities to utilize these CTCs. So there are several articles wherein they have tried to see that these CTCs would help predict chemosensitivity, and our group also had published the clinical utility of circulating tumor-associated cells to predict and monitor chemo response in solid tumors. So you take either fresh tissue or blood and isolate CTCs and try to run the standard chemotherapeutic drugs and try to see something like our microbiological culture and sensitivity test. So you have some idea whether the patient would respond or not. So since these are live cells, you are recapitulating the microenvironment of the body. So that is how these are beneficial. And therefore, this was from the recent ASCO again, Journal of Clinical Oncology. There was an abstract from our group. So in addition, so if you can look at this, one of the pioneers in the liquid biopsy segment, Dr. Rolfo, he had shown that there are two models. Now, model one is for cancer interception. That is, you're trying to find out what is the benefit, how can the liquid biopsy help in the screening of cancers. And the second model is for metastasis interception. So if you can see the rates, the survival rates, they are dramatically, which is as expected, decreasing from screening stage. So if you can address this problem, nip it at the bud at the time of screening itself, this would be the best way forward for cancer. So it helps in early detection, diagnosis, prognostication, disease monitoring, and minimal residual disease following surgery. Also helps in predicting relapse and therapeutic resistance. Widespread implementation of liquid biopsy is hampered by technical and implementation challenges. There are gaps in education. People don't want to believe. I've heard so many speakers saying that something new comes up. There is some kind of skepticism, but things will change. There are gaps in education, awareness, and training. And most important, patient's voice needs to be heard. Just today, someone calls me and says, Madam, I have gone to X hospital, and when this technique is already there, why are the doctors not recommending? So I cannot say that there is. So when something new comes up, people are not ready to accept it. So patient's voice is very much important, and we need to hear that. So this is very useful even for us healthy people. Get into this EasyCheck 360, which looks at 30 cancers, the commoner cancers. It doesn't address the rare ones. And this is available now in India for our own population. And True Blood, of course, is for symptomatic patients. And you also have the chemosensitivity assays. So to conclude, there is a need for improving the understanding of molecular profiling of tissue and liquid biopsy. We need to make use of these non-invasive solutions, scale them up so that we provide access to patients and individuals in remote areas who cannot immediately reach their providers. And probably when you scale them, the cost would come down. Now, molecular profiling has become routine than a last resort in oncology, and therefore emphasis on quality, accessibility to clinical trials, and insurance coverage may help adoption on a larger scale. So I thank my team at Datar Cancer Genetics, the 300 people who have really worked hard to reach this. There is a long way to go. We need to do for a lot more. Thank you all.
Thank you. Thank you, Dr. Sudha.
Thank you, ma'am.
Madam Kaiser Jameel, are you online?
No, I think Dr. Jameel had a problem, so she excused herself because of some internet problems.
Thank you so much then, Dr. Sudha Murthy. We'll look for, there's a lot of awareness should come, even among the clinicians and
Could I just ask one question briefly?
Right now.
Yeah, Sudha, when you're talking about screening, and for especially for prostate, then can it be ordered as a direct to consumer? Can anybody opt for it?
Yes, it is available as a direct to consumer. So there is an Easy Check 360, which looks at 30 cancers, as I said. There's an Easy Check prostate, which looks, but this is not US FDA designation status is not there for the.
That doesn't matter. The question nowadays, mostly companies do, in your TRF, you expect a doctor's signature. But for these, you don't think they need it, correct?
For the screening test, it's not needed.
Okay, thank you.
Is
You
it
can
for
make an offer it to patients then.
colon cancer also is available in the panel?
Yes, ma'am, it is. It is. It is.
Okay.
So
Thank
can
you.
we ask some very, very basic questions?
Yes.
So I understand that if the test is positive, we get a data. If it is negative, does it rule out?
At that point, when you have got tested, it does rule out. But then, like an annual screening, it has to be done annually.
Yeah. So there could be false negatives. So that's one thing we need to.
Yes. So fortunately, I think US FDA has looked at the entire data. There is a significant population of, as I had shown in one of my slides, asymptomatic people also have been tested. Normal people like you and me have been tested, and there was 0 false positive. So it is a highly specific test. Sensitivity is not as much. It is not.
Yeah, I'm not worried about false positive. And because false negative is an issue, not false positive.
Yes.
The other question is, theoretically, we believe that all the circulating tumor cells are not capable of metastasis because all the policemen in the body are sitting at every spot to kill them. So how significant is the mere presence of circulating DNA predict a metastasis? How does it go parallel? Because in the Robbins diagram itself, we tell about 10 checkpoints, how, metastasis is a tough war that a tumor cell has to fight. And so that's the last missing link, that's my always, it's not that I don't believe that, but this is some academic query that I have.
Yes, absolutely. So I think good that you brought this out. We are aware that the circulating tumor cells are there in the circulation or in the bone marrow, but they remain dormant in some. There was an big article by Dr. Pantel in Nature, where it remains dormant for nearly decades. So you have identified, it only says that you please follow up, you have a tendency. So we are not saying that you react to that and start doing it. So many people say, no, that will put me into apprehension and I cannot. So is it better to know or not to know? That would be the question, rather than saying. So as you said, not all tumors might really secrete into the circulation, but a significant proportion. So that's what our data says, that we have not been able to pick up even in one healthy individual. But we have picked up even in DCIS. We have picked up in DCIS, and by standard definition, we think DCIS may have, so it is an invasive cancer, which does not happen.
Well, every tumor now is a systemic disease. That's the reason, it's practically, whether we see it as a specific metastasis, yes, that's the increasing trend to believe. But how capable they are of metastasizing is one thing. The other thing is the one which goes to about the cancer stem cells, So it's believed that the non-CSCs probably will get removed, and the CSCs, as you mentioned about being dormant, and they are very uniquely competent to stay dormant, and they would emerge only when the opportunity comes.
Yes.
And specifically with the immune therapy coming up and all these things coming up, so it's the immune system plays a very big role in controlling these circulating tumor cells. So, which makes it quite unpredictable. Unless we have, not from normal population, I'm not worried about the normal population, but from the entire spectrum of the clinical spectrum. And the other thing is, do we have adequate number of patients' data? Because the larger screening you do, the more robust you would be your data.
Yes.
So from all stages. See, the thing is, we get data, but we are not sure whether this data, how does it match with the clinical staging or the grade that we are familiar with. So the matching data doesn't come neither in papers nor in, that's the limitation according to me.
I understand. So only larger number of multi-centric trials would really help instead of saying only one center is doing it. That's what we have tried to do, but then we have taken from US, UK and India. And this is a cohort of 60,000 patients for the screening. I mean, the patients and the asymptomatic healthy individuals. So similarly for the prostate. So as you rightly pointed out, there need to be more studies, and there are many researchers who are working on this.
And how about familial cancers, like, some colon cancers and all? Can they be predicted in the family, the next...
So CTCs do not address prediction. I am sure Madam Annie Hassan ma'am would be able to answer this. Hereditary cancer panels we do for the genetic, but it does not ensure that yes, this person is going to get it. It would not predict for sure that this person is going to get cancer.
Maybe those people, Sudha, maybe those people could use it for early screening.
Yes,
correct. BRCA1 carriers or Lynch syndrome. When we know there is, then the family members can be put on an earlier screening.
Yes.
That will be very useful. Yeah.
Instead of doing mammograms, which themselves are causing issues, they can be put on this early screening panel.
Radiation and all attended.
So, Dr. Sudha, did you work with any of these uterine cancer related things? Because what happens is most of the women are positive for fibroids. So when they have the fibroids, if the size is growing, they don't know whether it is the malignancy or it's simple, the fibroid is growing, hormone based or any such thing.
Benign.
So, benign. So can we differentiate based on these circulating tumor cells? Have you worked on that?
Yes, we have a solution which talks about screening of the female uterine organs. It's called FemSafe, Easy Check FemSafe, which looks at ovary, breast, uterus, and cervix. All these 4 organs. But in addition, as you said, if the patient is symptomatic, has a lump, has a fibroid or something, you have to do the true blood solution, which looks at the organ-specific markers in that particular, depending on whatever the scanning has given you. So this is very useful and handy to gynecologists, especially where they don't have access to frozen section. This test tells them that this is malignant. You can do a staging lab for a ovarian cancer, something like that. So
that is through hysteroscopy. That is through hysteroscopy?
Sorry, that is?
Hysteroscopic analysis?
Hysteroscope, I mean, whatever is the tool that is used for the surgery is different. This is only a simple blood test. So if the circulating tumor cells are there, they will be picked up and you can say that these are malignant or non-malignant.
So hysteroscopic diagnosis is not required.
Sorry?
Inpatient hysteroscopic diagnosis is not required.
We would not say at this point in time, it's not part of the guidelines. I'm foreseeing it very soon coming into the guidelines. But we say that at this point, standard of care has to be done, whatever is the gynecologist or the oncologist is planning.
Thank you, ma'am.
Thank you.
Can we come to now conclude the thing, Dr. Geeta?
Excellent talks. And I'm sorry about the technical glitches. I'm sorry for the technical glitches, but the content and the presentations were amazing. And the progress that all of us have made is something that, makes us proud of. And I'm so glad that I concentrated on picking up all the health technologists, actually, I would say. And for also crossing the boundaries, for getting out of the comfort zone. I must say this, when we go from one specialization to another, we feel that we are at the bottom of the ladder. And to catch up with the other people in that field is not easy. So, and with that, I really must compliment Sudha. And Dr. Annie has made it so, simple. Dr. Geeta's experience was visible in her slides, though I have to go through all that. And so I.
So I also
And
thank.
Priya, of course, Priya is the, really is the she makes the country proud, I would say.
Yeah,
With
yeah.
contribution and dot on time, the way they have been chasing. In fact, the corona has been changing us. They have been chasing the virus so well that, they didn't leave any stone unturned. So with that, I would thank all the chairpersons, moderators for chairing this, all the speakers and the participants for the patient listening. And the last is the best. I wouldn't say, but I mean, it's with no other thing other than that we are going to into the space now.
So, Geeta, I also would like to thank the speakers once again for their excellent presentations and all the delegates for their participation. I also thank Dr. Kaiser Jameel for moderating the session. I profusely thank you, Dr. Geeta Vemukanti, and all your team and the organizers and the IWSA team for giving me this opportunity to chair the session and to be a part of this memorable event. Thank you, one and all.
Thank you so much. Yeah.
Yeah, it was a good
Rita, over to
session.
you to chair the session. So Rita is known to all of us. In the interest of time, I would just say she's one of the fantastic radiation biologists. And in fact, if things had been better, non-COVID, maybe we could have started the MSc Radiation Biology course with her. But we took a backseat. So, and I would hand over the, mic to floor to Dr. Rita. Kindly moderate it and chair the session, please.
Yeah, thank you very much. Good evening to all of you who have been through the session from 2 p.m. today. And wonderful talks, wonderful sessions, glitches, yes, we learn all the time. Google Meet, I think this link of Google Meet may be having a little bit of a problem in the university and hence these glitches. And very good morning to my friend scientist, Dr. Sharma Bhattacharya. She has woken up at 4 a.m. in the morning from California to be with us and give her keynote address in this very special inaugural meeting, which the Hyderabad branch of Indian Women Scientists Association has organized. We have 11 branches. It's only for the sake of Sharmila, I repeat a little bit of what Dr. Lalita Dhareshwar, our immediate past president, had already said during the inauguration. Indian Women Scientists Association is turning 50 in June 13, 2023. We shall have an annual, a year-long programs to take our messages to the society, as has been our mandate, to take the science and scientific thought processes to students in the society across the board of ages. So this Hyderabad branch, one of our 11 branches, came up first with this program. So today we actually have launched a scientific session for celebrating Golden Jubilee year of Indian Women Scientists Association. The rest is all in the IWSA.net. And of course, when we communicate, you may recall when I met you in the past, I had always mentioned, because IWSA is like the next breath we all have. So whenever we meet somebody new, we definitely speak about IWSA. Now I get to the pleasure of introducing my friend scientist. She's Dr. Sharma Bhattacharya. The present position, she's a subject expert in the UN Senate Committee, which looks after commerce, science, and transportation. She has a career of 22 years with the NASA, where she served at various positions as principal scientist, chief scientist, only gathering experiences in missions, in taking biosciences to the space, and many more, which she will talk about during the address. She stands to me as an ambassador of integrated sciences, a career which can be shaped when you have integrated sciences in your study path. She began from the presidency alumnus as BSc degree in human physiology, in Kolkata, then moved shores to study biological chemistry, then molecular biology, then neurobiology, where she picked up the organism Drosophila. And all this together has given her what she has contributed to the society and science in the space biology laboratory, later the bioscience division of NASA. Sharmila, we are all awaiting to hear from you. And all my colleagues here, and on behalf of IWSA, an open invitation, you become our NRI member. Of course, I know. And thanks for gracing this occasion to give your keynote address. And as you have desired, there would be a question-answer session. And I will take one more introduction and request Dr. Sudha Padhi, a senior-most founder member who has joined to listen to this keynote session. Of course, a full day would have been tiring, so Madam has come and attended a few. Sudha Padhi, Madam, if you would kindly show the video and we all see you and say a very good morning and evening from the shores of West America and the India. I have my board of trustee members, Dr. Surekha Jhingre here, and many more members who were there, but I think the evening time, we have taken some extra, so they are all moved out. With the blessings of Sudha Padhi Madam all the time, I give the forum to Sharmila. And there's Sunita Mahajan Madam, another senior most madam of our IWSA. Computer scientists and physicists, and Surekha Jhingre is a protein biochemist, and every other person you know here is an eminent scientist in their careers in it. Okay, all yours. Thank you very much for giving me this time, Geeta, another friend.
Thank you so much. I mean, last time we had her 2 years ago, and I really wish she comes again. And I had visited her lab, and it was wonderful. Met all her students, and it was wonderful. Over to you, Rita. Sharma.
Thank you all so much. So firstly, good morning for me, good evening for you. It's wonderful to meet you all. Thank you, Rita, for that very kind introduction. Thank you, Geeta, for the invitation. And so there's definitely going to be a little bit of a switch in topics because we're going to talk about biology in space. So let me begin by sharing my presentation with you. And what Geeta and I figured out last night is just so that I can actually see all of you, I'm going to leave my slides in this mode so that I can see you. Otherwise, when I do full screen, I lose sight of everybody other than the screen. So I'm going to make sure that I keep my talk to about 15 minutes to leave enough time for Q&A at the end of my talk. So let me start by actually move to my first slide. So when we talk about space flight, there are some specific stressors that we deal with in the space environment, which is very unique to being in space and very different from what, Earth-based living organisms typically are exposed to. So one of them is, of course, reduced gravity, right? So for Earth-based systems, when you're put into space for periods of time, being exposed to either microgravity or reduced gravity. For example, if you're on the surface of the moon, you have reduced gravity. If you're inside the International Space Station, you have microgravity that you experience. Another aspect of this is you have ionizing radiation. Now, many of you are experts in the radiation arena in terms of cancer and so on. The radiation that you are exposed to in the space environment is a little bit different. It can be a little bit different. It's a mixture of heavy ions, which is part of the galactic cosmic radiation, like, iron, carbon, silicon, etc. Charged particles, which can really damage tissue. High LET radiation that can damage tissue as it enters biological systems. There's also protons coming from the sun, solar particle events. Those are a little bit more easy to shield against, but the heavy ions coming from galactic cosmic radiation is much harder to shield against. Along with that, we have I'm going to skip to this one. You have elevated temperatures. You have reduced temperatures. So sometimes when you're on the surface of the moon, for example, you might experience a 300 degrees Celsius flux in temperature between high and low temperature between lunar day and night. You're also in an enclosed environment and a habitat, so you can have almost 10 times more concentration of carbon dioxide than you do on the surface of the Earth. So a lot of environmental parameter differences in that environment. Similarly, depending on your location, you can be quite far away. Even on the ISS, you're still, a day or two away from coming back to Earth. When you're on the moon, you're weeks away from coming back to Earth. When you get to Mars, you're going to be, 2 to 3 years from a round trip to Earth. And so because of this distance, one has to really understand what this environment will do to biological system in preparation for these missions, so that you can take what you need with you, the medicines, the countermeasures, the screening, all of what you need for long-term exposure to space is stuff you have to take with you. And so the more we understand with research up front, the more prepared we are. And then lastly, there's the issue of regolith or lunar dust, which normally you are in an enclosed environment and you're protected from this dust. But when you do these extravehicular activities, when you go out in a spacesuit on the surface, of these, terrestrial. Yes. Was there a question?
No.
Okay. Maybe there's an open mic. So when you're exposed to these then, and then when you bring, when you come back into your enclosed habitat, sometimes these regolith particles or these dust particles, which are very, very fine, with static, they can actually cling to the suits. They can be highly abrasive. And so if breathed in, can cause issues in the lung and so on. And so there are all these multiple stressors that a biological system will experience when you get to space. And so one of the things that I'm interested in then is looking at the effects of some of these parameters on biological systems and having biomarkers and an understanding of the changes to anticipate. So one of the things that I wanted to bring up is, and if you just start by looking on the right side here, this was a paper published earlier that we then leveraged to look at some data. So if you look on the right side, essentially, what was done is that astronaut blood samples were taken before a mission and after a 6-month stay in space. And what you see is when you look at the lymphocytes and you look at the chromosomes and you start to look at the change in frequency of chromosomal aberrations of one kind or the other in these lymphocyte cells, you see that there is a statistically significant increase in the frequency of these chromosomal abnormalities taken from the lymphocytes of blood from astronauts who have returned from space 6 months later. And so, of course, the samples are taken from the same set of astronauts before and after a mission to do the comparison. And the thinking there is that this increase in DNA damage is 80% of that comes from galactic cosmic radiation, which are very difficult to shield against when you're in space. And so the work that we did is to then look at some of those endpoints, like, for example, translocations in the lymphocytes. And what you see here in the dark circles is the data from International Space Station, which is still relatively low orbit and therefore protected by Earth's magnetic shell or magnetic sphere, also called the Van Allen belts. That protects to some extent, it protects to a large extent the surface of the Earth, but it also protects to some extent the International Space Station, which is roughly 350 kilometers above the surface of Earth. It's orbiting at that altitude. And so if you look over time, you see that there is of course, the longer you are in space, there is an increased, probability that you will have damage to your DNA. But look what happens when you start to predict what will happen in deep space, beyond the Van Allen belts. That means when you're on the surface of the moon or you're on your journey to Mars, and you spend extended periods of time in those environments, you can see that the estimated translocations per cell starts to increase quite dramatically. Now, there is obviously a large uncertainty because so these are now done, this data that we had was entirely from predictions and estimates made based on the anticipated radiation environment. But the things that we actually don't know, which is what we need to test, is that in addition to the radiation, as I mentioned earlier, you have other stressors like reduced gravity. And we know from previous studies, and I won't have time today to show you all of the data and all of the publications that we have, so I'm going to just hit on a few of the highlights. But when you look, for example, at oxidative stress markers, gravity alone, can really increase the oxidative stress markers. So then when you couple that with radiation, which also causes oxidative stress in the system, you can imagine that the combination of the two can significantly increase some of these endpoints that we can measure. And so that's what makes it, for us, interesting to begin to really assess some of this. So the next slide here then, I'm showing you one of the instruments that we developed to start to anticipate what the extent of DNA damage will be on living biological systems when they're exposed to these levels of radiation for a somewhat extended period of time beyond the Van Allen belts, beyond our protective magnetic shell around us, around Earth. And so this is a mission called Biosentinel. I started as PI of this experiment, and then lately, as Rita and Geeta pointed out, I've moved to Washington, D.C. to take on some other roles. I'm actually currently the program scientist for space biology at NASA headquarters. And so now I've transitioned some of these duties on to my deputy, who is now the lead scientist for this mission. And it's very exciting because it's actually scheduled to fly in the next few months. And I'll tell you about the rocket on which it's going to fly. So this experiment we developed these microfluidic cards to house yeast cerevisiae cells, eukaryotic cells, whose DNA damage and repair mechanisms are similar to humans. And what we find, and what we do actually, is we use these microfluidic cells with the yeast cells. We have optical detection systems with LED light that when it passes through these microfluidic systems, and you see that over here, you have many of these microfluidic cards with channels with these yeast cells in them, we use dyes like Alamar Blue, which are oxidative, redox sensitive, so that we can monitor the redox state of the cells. We also have different wavelengths of these LED lights so that we can measure growth and assess the growth rates, etc., as the cells get damaged. And actually, this experiment will be done in this satellite. This experiment is being done in a free-flying satellite. It will actually eventually go around the sun. And of course, as it goes further away from the Earth, it's harder and harder to get the data back. This experiment that we developed and the technology that we developed is entirely for being able to do this experiment in space. The samples never come back. So all of the experiment is done by the data being telemetered back to Earth. And let me see what else. And so what this experiment will allow us to do then is to quantify the rate of DNA damage in these cereBCI cells as the cells, and we're going to fly wild type as well as mutant lines that are more sensitive to radiation damage, in the homologous recombination pathway, and then get that data back and analyze the effects of the change. And so this experiment, this hardware, it's basically a 6-unit CubeSat box. So it's roughly the size of a bread box. And we're very excited because it is a secondary payload that will fly on the first validation mission, the first test flight of the large rocket, which is called the Space Launch System, SLS, which its maiden voyage to test the heavy lift capability to take science as well as people, on the crew vehicle, on the Orion. Crew capsule is where the astronauts and cosmonauts, etc., can be housed. And then there's a stage where our experiment will be one of these boxes that you show over here. And when the rocket goes into space and the crew vehicle gets detached and then comes back to Earth, our experiment is then shot out into space, and it goes in a heliocentric orbit, immediately begins to activate its batteries, etc. And so it's a completely autonomous experiment that we will then do in space and send the data back by telemetry. And this experiment is scheduled to fly this summer. We'll see exactly what the launch date is, but our experiment is already, prepared and loaded up to fly on this particular mission. So now I want to shift gears a little to another experiment that we did some years back on the space shuttle. As you know, the space shuttle now has been retired, but when I first started at NASA 23 years ago, I guess, that was the workhorse to do science, was the space shuttle. And so in that particular case, we used the Drosophila model, the fruit fly model. And as Rita pointed out earlier, I've worked with a number of different model systems through my science career, and worked also very closely with engineers. And so this multidisciplinary approach to science is one that I really appreciated doing and enjoyed doing through my career. And so what you see here are systems that we had to develop with partnership with a lot of different groups domestically and internationally to look at multi-generational growth of Drosophila melanogaster as a model. Including, systems like a video camera system, so we could monitor the health and behavior of the flies in space, as well as specific modules that the crew could help change out so that we can separate the different generations and look at the changes that space imparts on these systems. And so this publication that we had looked at the immune changes of the Drosophila model in space. And some of the things that we found, for example, is that when you look at the number of plasmatocytes, so this category of the major category of blood cells in Drosophila, we've actually found a reduction in the number of plasmatocytes in the animals that had bred entirely in space. So these are for the offspring of the parent flies that we sent into space, and then the offspring that were born and developed entirely in space. When we brought these larvae back and we looked at the blood system, the number of these blood cells were reduced. And what we also found was that concomitantly with that, there was also a change in the size of the larva. So there was a reduction in the size. So these again are the ways in which we're beginning to get a sense of what are some of the changes that we can anticipate. And then, this was still in the day when Affymetrix chips were the way that we did a lot of our genomic analyses now, and the transcriptomic analyses. Now, of course, in the last several publications in our lab, we're using RNA-seq methodology to look at changes. But this, just to give you an example, that when we did those microarrays, what we found is that there was a significant increase in the expression of humoral innate immunity markers, blood cell maturation markers were altered, and there was changes in phagocytosis function of the blood cells. So then to look at function of the blood cells, since we had already gotten an indication that there was a change in these plasmatocytes, in terms of number and in terms of markers, genetic markers, or the expression of these genetic markers. So then we started looking at function of these phagocytosing plasmatocytes. And what we found is, excuse me, that there was also a change. If you compared the ground cells versus flight, there was a change in the efficiency with which these blood cells were able to phagocytose bacterial particles. So there was, from the animals that had come back from space, there was decrease in the efficiency. Similarly, when you look at when we looked at the number of bacterial cells that the plasmatocytes could engulf or capture, there was also found to be overall a reduction in the number of blood cells, in the number of bacterial cells that these blood cells could capture over time. So there was not only a change in structural components, as well as, omic, transcriptomic genomic changes. There was also change in the function of these innate immune markers in the animal. And so many of these innate immune changes then mirrored some of the changes that have been noticed also in humans post-flight, and begins to serve as a simple model where, remember that in spaceflight, the challenge is the sample size. And so when you use these surrogate models where you can fly large numbers, large populations of animals, you can see that these changes are subtle, but they're important. And so having a large population size for spaceflight studies become important. Now I wanted to highlight another experiment that we published recently using that similar hardware that I showed you, with the capability to do multi-generational studies and separate the generations in flight. So in this particular study, what we did is we exposed the bacteria Serratia marcescens, which is a human pathogen, but a particular strain, DB11, is also a pathogen for Drosophila. And what we did is we exposed the pathogen to the space environment to look at. Now, I showed you first that the immune system of the host animal or host organism alters as a result of spaceflight. Now we wanted to check what happens to the pathogen when it's exposed to spaceflight. And what we found is Serratia shows a significant increase in virulence. So when we brought the Serratia marcescens that had been exposed to spaceflight back to the ground, and we exposed flies with these bacteria that had been exposed to space, we found that there was a great increase in the lethality of the hosts that were infected with the space-flown bacteria when you compare to the ground-flown bacteria. Obviously, your controls are done in an identical hardware, identical environmental conditions. The only difference is one set of experiments was sent to space, the other was on the ground. And so there was an increase in virulence. What was very interesting also, and we have a subsequent paper that we're currently working on. We're doing revisions to the manuscript for resubmission to the journal. What we're finding is that these changes actually in the bacteria then can get reversed. So as you grow these bacterial cultures on the ground, you can recover, the old properties of the bacterial cells. And so these are transient changes that the bacteria seems to incorporate as we take them into space and then bring them back. So this was another assessment that we wanted to do of the changes brought about on microbial systems that could be exposed to the space environment. Now, my lab also utilized mammalian systems. So we remember another big thing for astronauts is to look at markers, easily detectable markers, for example, in the blood, that could tell us a little bit about the status of how things are going during flight. And so we looked at the neutrophil to lymphocyte ratio in mammalian systems. We used rodents. We used data from rodents that have been exposed to spaceflight. We used data from human beings that have been exposed to spaceflight. And then we also did ground-based studies, equivalent studies, so to speak, with human blood cells exposed to simulated microgravity on Earth in a machine called the rotating wall vessel. It basically causes a continuous suspension of the cells, so that basically the cells are in free fall continuously to sort of mimic the effect of microgravity that is experienced in space. So we did human cells in that system. And then lastly, we did hind limb unloading of mice, which is also often used to try and see that when you don't have gravity loading the hind section of the mouse, how does it affect things like the immune system and other parameters. And in all of these cases, whether they were blood samples taken from space or whether they were the ground-based analogs of spaceflight, these models that we used, in all of these cases, both in the rodents and in the human models, we found that there was specific times in flight when there was an elevation in the neutrophil to lymphocyte ratio, the NLR ratio, just as, again, a biomarker for a quick assessment of the effect of spaceflight. And then one more item that I was going to show you in the interest of time. I'm afraid I can't show you some of our neurobehavioral data and the brain data and so on, but I wanted to show you another set. This was a really simple set of hardware that we developed. It was a passive environment. It was a ventilated box that we used for Drosophila experiments to look at cardiovascular, changes in the cardiovascular system imposed by spaceflight. And so this was a paper we also published recently, which looked at so what you're seeing here on the left side is both sides actually, is the heart. This tubular organ is the heart of the Drosophila, of the fruit fly. And marked in red are the myofibrils, structure, which, you can see that it is in the ground controls. These myofibrils are very circumferentially arranged so that when they contract, it allows the blood in the Drosophila to flow in this in the heart system in one direction. And when you look at that same organ from an animal that has developed, that was born and bred in space and brought back, what you see, and in red are the myofibrils, and you can see how disorganized the structure of the myofibrils is in this heart organ. And in green is the extracellular matrix, which is, associated with the myofibrils. And you can see that there's a dramatic change in morphology and structure of both the extracellular matrix and the myofibrils in the animals coming back from space. And so these are the structural changes we found. And this was a study done in collaboration with my partners and collaborators at the Sanford Burnham Research Institution, Discovery Institution in La Jolla, in San Diego, California. And basically, as you can imagine, that when you see such a change in structure, you also get a change in function. And so we looked at contractility, and the hearts were smaller, they were less contractile. And essentially, and we also looked at proteomic changes in this publication you'll find. So tissue remodeling and proteostatic stress seems to be a response that we see in these heart muscles, and we are beginning to see in other organ systems as well from these space-flown animals. So then, to summarize, the space environment comprises many stressors that can affect biological systems, including humans, that are exposed to this unique environment. And some of the stressors that are of particular note to us is this level, elevated levels of ionizing radiation and changes in gravity levels. I also talked about how we can use multiple biological model systems as surrogates to probe the effect of spaceflight on human health. And I showed you examples from immune studies, cardiovascular studies, et cetera, that we have done, as well as pathogenicity of microbes. I didn't have time to show you the neurobehavioral changes that we've been looking at as well. And also the fact that one can use, depending on what you're studying, a variety of different model systems, which give you that advantage of statistical size of your samples in doing spaceflight experiments. Because, contrary to how it sounds, space is actually limiting in space. So when you do spaceflight experiments, there's a limit to the size, the volume, the mass of the payload or the experiment that you can send into space. And so having these surrogate models like Saccharomyces cerevisiae, Drosophila melanogaster, mammalian cell cultures, rodent models, these make it easier then for us to begin to look at the detailed physiological changes in these systems. And then, as per the theme of this workshop that you have all organized, then the idea, to do science here in space, we need to develop the technology. And so the requisite habitats, the measurement instruments, things that I, in fact, didn't have time to show you. There are many instruments, including PCR machines, bone densitometry, measurements, instruments, radiation sensors, blood pressure monitors, heart function monitors. These are all things that are developed, and in order to use them in space, they often have to be highly automated. They have to be miniaturized, and the data often needs to be sent back to Earth by data telemetry rather than having to bring back, hard drives or instruments back down. And so these are all technologies that one then has to adapt or develop for the first time for use in space. And these then become very important tools for us to conduct research in space. And with that, I will stop and thank you very much for your attention and take questions as well.
Thank you so much.
Yes, absolutely. And I'll stop presenting so that we can free up this. There we go.
I would request the moderator to please carry on with the session of question answers. There are a few comments in the chat box.
Dr. Sharmila, but one question I have is, have you looked at the effect of these rays on germ cells?
So stem cells and germline cells is what you're thinking?
Germline cells, yeah.
Yes. So, we are not specifically working on that, but you're absolutely right. Dr. Akka, this is an area that is being looked at. So, for example, there are groups like Dr. Tash, for example, is looking at germ cells in mice. And there have been changes found. Oh, and yes, another example from Drosophila, for example, where, it's a really good model to look at reproductory cells because, again, you can do large numbers. And so there was an experiment done by a Japanese group a few years ago where they looked at the effect of radiation on the germ cells. And what they did is they took males into space, brought them back down, and then they did an F2 analysis, to look at transmission of the X chromosome linked damage. And what they found is, as one would expect, that there was a significant increase in the X chromosome damage in the germline cells from the sperm of the males who had been exposed to space. And the interesting thing is, I forget exactly, but the males were exposed to space life for only about 9 or 10 days. It was a shuttle mission, so it was a relatively short period of time. And in spite of that, there was a significant spike in germ cell DNA damage. So yeah, great question. Definitely, that's an area of interest.
Thank you.
I saw Dr. Surekha's followed by Annie.
Yeah, thank you.
Please, short questions, please, in the interest of time.
Just a continuation of this question raised by Dr. Jyoti. You mentioned earlier that some chromosomal aberrations were seen in the astronauts who went abroad to space earlier. Are they being followed up? I mean, is there a routine follow-up to see what is the change or what are the changes that are being are coming up, should we say? It may not be immediate, it may be a long-term effect.
Exactly. Yes, absolutely. Dr. Surekha, it is indeed being done. And in fact, what happens is the astronauts go through, astronauts, cosmonauts, from all over the world, the same protocols are observed. So there are very detailed sample blood and urine and all kinds of analyses, and samples are taken pre-flight and post-flight. And then longitudinally, they are monitored post-flight, definitely for like a year or two following flight. But also, you bring up a very good question. There are publications from the long-term effects, because many of you are interested in the question of cancer. And there has been long duration studies done with astronaut cohorts. Remember, the two caveats, of course, are sample size, because again, there haven't been that many astronauts. When you compare to your patient populations, the relative number of astronauts is still small. However, in spite of that, there is seen to be changes in the long duration. And the second caveat there is that the astronaut pool is a particularly healthy, well-trained, physiologically robust group, a population, which also makes them different from the average human population. So there is also that caveat. But in spite of those caveats, yes, there has been shown to be an uptick in the in the long-term effects in terms of cancer. But let me also, your question is a really good one. Another thing that it raises, there are even short-term changes. For example, if you look at the brain, if you look at changes in neuronal structure and function using let's say a rodent system, let's say you do radiation exposure, and you can do this on ground-based studies, and you simulate space radiation over a period of a few days, and then you look 9 months later in a rodent, you see persistent changes and permanent changes sometimes in brain structure and function, so central nervous system changes. So yes, and similar things have been looked at for cardiovascular function, like vasodilation. There are changes, from radiation and Heinlein unloading. Some of these are ground-based studies, which simulate spaceflight. And then 9 months later, you still see persistent changes. So yes, there are both short and long-term changes as a result of radiation.
Thank you so
Good
much.
evening,
And
ma'am.
Annie,
something going up in the air.
sorry, one question each. So Annie followed by Chitraji.
Yeah. Hello. Yeah. I just wanted to know whether epigenetic studies have been done, because there are multiple things you showed in the environment. So have they been done?
Yes, they have indeed been done. And there are found to be epigenetic changes. And you know what's very interesting is they are looking at epigenetic changes in mammalian systems as well as in plants. And they're finding that there are actually epigenetic changes occurring across the board. So it's not just in animal systems, but also in plant systems. There are, and yes, there are studies being done by the community along those lines. So yeah, another great question.
Chitraji.
Yeah, maybe it's a very basic question, ma'am. But
No
in
problem.
female astronauts, is there a change in their reproductive system when they are exposed to the space?
Very good question again, Dr. Chitra. And so here's the thing. So when we talk about, and as earlier, I think Dr. Jyoti, was asking about this question of, germs, changes in germline cells and, multi-generation effects, which, again, epigenetic changes can also, cause some of these changes. Now, in astronauts, remember that the relative exposure to the space environment, if you look at their life cycle, is relatively short, right? And also, having said that, with astronauts, what they do try and do is that they do try and have astronauts who have who are not planning to, for example, have kids after doing like sustained, reproduce after sustained space flight for long periods of time, right? So there is consideration given to these factors, because the thing about radiation in space is that it's completely stochastic. You don't know which of your cells are going to get hit at what frequency or when. So there is a large uncertainty, yes, but that probability is significant that reproductive cells could get hit, with a large error bar, because again, there's this large uncertainty. And so, yes, the bottom line answer to your question is it is a possibility, and therefore steps are taken to ensure that it doesn't, impede the functionality and the next generation. Exactly.
Dr. Sudhika, you had one more question? Or your hand is still raised?
Geeta.
I have one question.
Yes.
I have one
Geeta,
question.
I had, Dr. Geeta, are you going? I had one question about stratification. So when we talk about 2D and 3D cell cultures, my guess is without gravity, you may have a 3D construct in a much more easier fashion than what we see. So there was a paper on NASA bioreactor for retinal multi-layering. So is that of interest to you?
Yes, absolutely. In fact, there's one of my colleagues, in fact, who is, very interested in these areas. And now, in fact, we have a call, a solicitation, a proposal call that's been out to get research, in this area of 3D tissues and microphysiological systems to look at the effect of spaceflight. So you're absolutely right, Geeta. It is a useful model where you can use, human cells in a 3D environment to look at the effects of spaceflight. And Dr. Sharma, I think you had a question, but I think you might have had a problem with your microphone.
You're muted.
Yes, we can't hear you, Dr. Sharma.
I have a question. Am I audible?
Yes, I can hear you.
Yeah, I am. Yeah, I'm Manjushri here. My question is, what is the impact of all this radiation as well as the microgravity on plants?
Yes. So we have, so now that I'm sort of managing the science for space biology at headquarters, half our portfolio is actually animal biology and the other half is plant biology. And the effect on plants is also of great interest to us. Because as you can imagine, for humans to thrive in space, we're going to need, in the long term, to have plants thrive in space as well. So in the International Space Station over the last 10 years or so, we have made quite a bit of progress, not just from the NASA side, but from ESA, from other countries as well, where we have been growing plants in space. The challenges for growing plants in space, where we talk about technology development as part of this workshop, some of the challenges is the watering system, for example. And you can imagine on Earth, gravity plays such a big role in watering, so that, the fact that, gravity is pulling water down, the plant has to have specific systems in which to bring water up from the ground, right, for the stems and the shoot systems. But in space flight, where you don't have that gravitational gradient, it actually makes it, contrary to what you would think, it actually makes it very difficult to develop watering systems that are very effective. So we do have some, and we have tested them on the International Space Station. We've grown things like lettuce and Arabidopsis, of course, because that's a workhorse as a model plant. Many other crop plants like, radish and peppers and so on and so forth. Like I mentioned, folks have looked at omics changes. So there are transcriptomics and proteomic changes, as well as, epigenetic changes that have been noticed in plant systems, that have been developed in space. There have also been multiple, there have been efforts to replant in space and look at that. It's still definitely something that we're working out to make more and more efficient. And as you can imagine, eventually we're going to need large numbers of plants. And again, as we were saying, space and volume is always a limiting factor, and mass for taking things to space. So right now, all of that is being done in relatively small habitats. But yes, plants have been successfully grown. There have been changes, that folks have looked at. There are other studies you will find in the literature for plants, looking at the effects of different wavelengths of light on the growth of plants in space. There are other papers looking, for example, at, lignification of plants. And if you reduce lignification, which on Earth, the plant would not be able to support itself because it has to support itself against gravity. And lignin sort of forms a skeleton, for the plant to stay upright on the ground. So there are scientists who are looking at reduced lignin plants to see if that actually helps the amount of plant material that is consumable by humans, because the lignin part is the part that's not usable usually. And they're looking to see what is the effect of these reduced lignin plants in space. Can they grow normally and so on? And some of that data is in the literature, and it's all quite interesting. But yes, long story short, plants are being looked at. There are changes. And we are developing technology to grow plants effectively in space.
Yeah, I think by 2024, ISRO is going to launch its first human-made space mission.
Exactly. In fact,
to launch. Yeah.
Yes, in fact, a couple of my students who've worked with me in my lab in California have actually come back to India and are working in some of the space areas, including my postdoctoral fellow, Ravi Hosamani, and then Siddharth Pandey was, he was working in Bombay for a little while as well. I think some of them may be coming back to the US again to do research. But yes, so I've been a little bit in touch with ISRO's plans based on that. Yes. So good luck. I'm excited. I think the more we have, more nations getting involved in this, the more data we'll have, more research we'll do.
Yeah. What I wanted to ask was, can you hear me now?
Yes, we can.
About plants and more specifically succulents, they need a lot of sunlight. And if you're going closer to the solar area, then is there any variation in their growth? Because even if you compare the hottest place in India versus some places in US, they flourish so well over there as versus here.
Yes, yes, yes. Interesting. So the thing with, at least so far, with plants, what we're doing is, plants and animals, we are growing it in our protective habitats, inside the International Space Station. So in some ways, there is increased radiation exposure, yes, because like the galactic cosmic radiation, like we're saying, is very hard to shield against. So it comes through the shell of the of the International Space Station, the aluminum structures. But for the sun, for either the UV or even the protons, which are coming from the solar particle events, those get shielded when you're inside the structure. So we are not, able to really distinguish, the particular kinds of radiation we will get from the sun directly from the ISS studies. Having said that, we actually currently have a call, a solicitation for proposals that we put out for plant biology, where we have specifically asked to look at the effects of elevated ionizing radiation on plant growth, to see what are some, just like I was telling you in the descriptions with the animal systems, we know that there'll be change in, we were talking about germline cells. We know that there'll be change in DNA damage, and what therefore will be the physiological effects in a plant when they're in this elevated ionizing radiation. And so we have got a solicitation on the street now. And so within the next year, we will be selecting proposals to look at some of those changes based on elevated radiation, doing ground-based radiation from particle accelerator studies. So yeah, it's a great question. It's definitely something in our pipeline that we are working towards.
Thank you.
I know Dr. Surekha has, but she had an opportunity. If it's burning, then we can take it. If it can wait, because it's already nearing 8 now. Surekha? Is it a short question, or is it?
No, I didn't have another question. No, I didn't have.
Hand is still raised. Okay, thank you so much. Sharmila, there are no words to thank you. And especially morning, if you're so bright in the morning, so bright and energetic, and you stimulated all of us. And fantastic compliments for you. So I thank you, and I would now like to thank Rita for sharing the session, and my co-host. I would now request my co-host and Ratna and others from IWSA to conclude the session through their comments. So, before I give chance to the other persons in the session, I would like to. It's a pleasure, the protocol that I have to thank Doctors Rita Mukhopadhyay, Sunita Mahajan, Dr. Lalita from Mumbai headquarters for giving us the first opportunity to be a part of the celebrations of the Golden Jubilee of IWSA. And I also thank our Vice Chancellor for gracing the occasion and for giving us his valuable comments. I would like to thank Dr. Ratna, Dr. Lakshmi for playing a key role in making this webinar happen. And they have been in constant touch with Dr. Geeta and me, and they were answering all the questions, WhatsApp, email answers, very prompt. And my heartfelt thanks to them. And have a message to take back home.
The lecture is also very interactive. I would like to thank all the speakers, Dr. Anuradha, Uma, Geeta Pepperty, Shilpa, Manjushri, Priya Abraham, another speaker, Dr. Geeta Sharma, Dr. Sudhamurthy, and Dr. Sharma Bhattacharya. All speakers have really agreed to give lectures, and as I said, the lectures were very interactive. And special thanks to Dr. Sharma Bhattacharya for joining us at odd time for to make this happen. Thanks a lot, Sharma, for joining us. And I'd also like to place on record the this extended support that has been extended to us from all the support staff. Very
Yeah, we are losing the voice here.
Can I say something? I'd like to say something, Rita.
Yes, madam, I will bring you in after the speaker. She's not been audible after she is done.
I would also like to say something.
Unmute, please.
We can't hear anything.
Correct. The
Can't
Google
hear me.
link, no, the Google link was strongly weak. I mean, I use this word. It was really giving us a lot of trouble today.
Yeah. But we can hear you all.
Okay. So Geeta, yeah, I think there's a electronic interference of two sets from which the link has been opened.
So Shall I say something now?
Yeah, I think Geeta, I will request Dr. Sudha Padhi madam wants to make a comment, and we will
I would
listen
like
to
to
her.
congratulate IWSA Hyderabad for this excellent webinar. All the speakers had a lot of information to give in their respective topics and regarding the advancement made by technology in the medical healthcare sector. So, thank you, Madhu, for reminding me at the nick of time so that I could catch up with this seminar. And one more request, all of you eminent scientists, speakers, please join us in our endeavor to spread science amongst the humanity and all, because science and technology is very, very important in our lives and everybody we should know about it. Thank you and bye.
Bye.
So, madam, can I talk for a few minutes?
Yeah, Sunita madam, the validatory session begins.
First of all, I would like to thank you for inviting me to participate in this last session. And it was such a wonderful one, a wonderful start for the 50th year. And I must thank the young girl from California who has risen up and given such a wonderful lecture to us. All that was really new and it's very interesting. Another thing I would like to say about technical glitches, that we learned one lesson: we must persist, and the knowledge that poured after that was so wonderful. That Geeta Sharma and Dr. Annie and the last one, Sudha Murthy. It was really too much. And even the earlier session was really wonderful. From 2 o'clock till now, we don't know how the time passed. Thank you, Geeta, for arranging such a wonderful thing.
Yeah.
And as Padya madam suggested, all of you will join us for the final celebration and all that.
Yeah.
So the triennial meeting will happen in the June
I think I
of
won't talk
2023.
more about it because we are already very late. So thanks a lot, everyone. And one more thing I would like to suggest Hyderabad branch, that please try to have your own place as we have at IWSA headquarters, Working Women's Hostel and all that. See if you can have it, because that gives a base and a foundation, and then you can really soar high
Right, right.
on the wings of change. So, thank you once again.
Very true.
And I thank IWSA for giving us this opportunity, and especially being the first foreigners. So, so this was a starting trouble, but and you did so well. So with that, I thank everyone, especially the senior faculty who stayed long to encourage us and support us. Thank you all much. So with the permission, I will now.
Yeah.
Okay.
So we have actually come to the end of the session, and
yeah,
on behalf of Geeta, whom we are not being able to so clearly hear, Hyderabad branch, congratulations to having put the stepping stone forward. The next one comes Kalpakam, and then the Bangalore, then Roorkee, Delhi, Nellore, Nagpur, and Kolhapur, Amravati.
So
all
ball
of
has
these
started rolling.
luminaires, ball has started rolling. We will have various kinds of activities. One of the branches is going to do an Olympiad, an All India Olympiad for maths for children. We are asking if the Bangalore can do a space session and Kolhapur can do a marine session, so that we cover different aspects of science, engineering, and technology, and be the face at those cities. And if we can open up and have physical ones, then it will be very nice. Otherwise, we'll continue on the online forum. 23 June, that is exactly 1 year and 4 months away, we will be having the triennial international meet, for which the flyers would soon go out. So I would request all of you to help in for making that a success, like how the Hyderabad meeting, the last triennial, was an amazing success, including the cultural activities, Geeta. So with this, thanks from all of us at the headquarters, and wonderful day of science. Congratulations again.
Thank you.
Thank you. Thank you so much.
Bye and good night.
Bye. Bye. Have a good day, Sharmila.
Bye.
Yeah,
it was good